it's a normal cell line, human embryonic kidney cell line. So why we have taken this cell line? Because these cell lines are recommended by researchers to highly transpectable cell line, and in our case, the strong reason was that this IRF1 family members are not expressed in the this cell line. So and whatever the change we will get after transfecting with our DNA or gene, so that will be the only our gene, not from the internal or endogenous protein. So that's why we have taken this two cell line. This is normal cell line. This is transformed, means it is actually close to the cancerous cell lines. It has all property of the cancerous. So when transformed, means it may either form malignant cells or benign tissue. So this is a type of cancer, and this is normal cells. So normal cells divide. It has controlled cell division, where it has uncontrolled cell division. So first we optimize the transfection condition. We used a lipofectamine technique. This is the a type of lipid membrane we mixed with this vector. Actually, this is the control vector expressing the GFP protein, green fluorescence protein. So once we transformed it into the HEK293, HEK293T, then we observed under the fluorescence microscope. So this is the 48 hours. This is normal. And after 72 hours, we got the 80 to 85% transfection. Similarly, in the case of HEK293T cell, this is the now at 72 hours, we got the optimal transfection ability. So we fixed, also we used a gradient of the DNA concentration. So 4 microgram we have fixed for the transfection study, and 72 hours for the maximum time for the maximum transfectability. Now this is the experimental condition. The cells were grown in the DMEM media with 10% FPS. So this is control cell. No any DNA was added from the outside, and this is the mutant wild type IRF1. This is wild type. So after 72 hours, this is HEK293T. So we observed some changes in the cells, transformed cells. That cells numbers were decreasing. But in case of truncated IRF1, this is the red one, the truncated IRF1 pcDNA mammalian expression vector. So this is we did not check if you compare with 0 hour with 72 hours. So we did not see any change in the cells number. But in case of wild type IRF1, we observed that diminution, reducing the number of cells. So then it is the pcDNA control. Again, this was control, empty vector was used, and there was no change. So what we observed that there is a reduction of cells by wild type IRF1 in transformed cells. So it is actually, we hypothesize that the cells are dying, and dying due to apoptosis. So that we checked further. So truncated does not have showing any effect. So it has a DNA binding domain, but it lacks the activation domain. So it does not activate the other genes which is involved in the apoptosis or anti-apoptosis. Now again, we same experiment we repeated with the control normal cell line. This is the normal cell line. So again, we this is the normal cell line HEK293, and then this is pcDNA wild type. So again, there is no change. It's a normal cell. It is not showing any cytotoxic effect. But also similarly in the truncated, there is no cytotoxic effect. This is controlled pcDNA, so no cytotoxic effect. So this is the graph showing there is no change in IRF1 wild type or truncated IRF. So there is no any cytotoxic effect showed by IRF1 wild type and truncated IRF1 in normal cell. We then check the viability of cell. So we using MTT assay. So MTT assay is a assay in which tetrazolium dye is interact with the oxidoreductase of the enzyme of the mitochondria of the living cell, and convert the tetrazolium dye into the purple color furazan chemical. So it is done. It was done in the 96 well plate. So cells were seeded equal numbers of cells. Then we did this cells study by MDT-assay. So we observed that only wild type is showing the reducing the cell number in transformed cell in the cancerous cells, but not in the normal cells. So there is no change. But also we observed there is no change by truncated IRF1. So this is reducing up to 100 to 40% by wild type, which is the its tumor suppressor activity. Now further what we done that we then further we go in detail so that why this wild type is showing some cytotoxic effect, why cells are dying. So we selected some uh, genes which are exclusively involved in the apoptosis. Apoptosis is a programmed cell death, and it is what we observed that this is surviving. It's an anti-apoptotic protein. So when we did the Western blot, so we observed that there is a reduction of survivin protein in the HEK293 T cells line by wild type IRF, but not by truncated IRF1. Similarly, dissimilar to this, in the HEK293 normal cell, we did not observe any reduction of the anti-apoptotic survivin protein. So it means this protein is decreasing, means it is actually apoptosis is induced in the cancerous cell lines, but there is no change we have observed in the HEK293 cell line. We have taken other gene BCL2. It's a anti-apoptotic gene. So that we checked it. Again, same procedure, Western blot we have done. And then again, truncated IRF one did not show any change in the expression, but wild type is showing the reduction of this gene expression. So it means actually it is again anti-apoptotic. So it will reduce, means apoptotic activity will be induced. And BCL2 is a mitochondrial membrane protein. Absence of this mitochondrial membrane protein induces mitochondria to release the cytochrome C. So cytochrome C, once released, it interact with the caspase 9, by which it induces the cell death, or it degrades the DNA of the cell, and the different features like membrane bleaving is happens, and this is due to apoptosis. Similarly, similar to that, we did not see any change in the expression of this normal cell BCL2 gene in the by wild type and truncated IRF1. Now again, we checked cyclin D. Cyclin D gene is a cell cycle progression gene. It actually phosphorylates the retinoblastoma tumor suppressor gene. So it actually, normal cells, it controls the cell cycle regulation, and G1 to S progression it requires, absolutely requires. So we also checked it by Western blot. So we again found that the level is going down. So this is its level is increased in the cancerous cells, transformed cells. So again, this IRF1 wild type is down down regulate gene expression, but truncated is does not showing any change. Similarly, HEK293 there is no change. So it means wild type IRF1 is no cytotoxic effect, no any apoptotic effect on the normal cell, but it has apoptotic effect or inducing effect on the HEK293 T cells. So here is the conclusion of my study. The recombinant truncated GST human IRF1 was successfully generated by deleting 92 amino acids from C terminal of the human IRF1 by saving nested PCR. The recombinant truncated human GST IRF1 was expressed in E. coli BAL21 cells. The DNA binding activity of the E. coli expressed recombinant truncated IRF1 and BRE beta is the virus response element. Actually, this region is responsible where IRF interact of the interferon beta promoter. With were observed and concluded that DNA binding activity is comparable with the wild type IRF1. So binding activity of both wild type and mutant type IRF1 was comparable, but transcriptional activity was not comparable. The truncated IRF1 did not inhibit the proliferation and not induce death in HEK293 T cells as showed by wild type IRF1 by trypan and MTTSA. Western blot analysis of the protein from the HEK293 T cells showed that wild-type IRF1 activated the expression of IRF1 regulated genes such as cervivin, BCL2, cyclin D, D1 at the protein level, whereas expression of the truncated IRF1 did not activate the genes in HEK293 and HEK293 T cells. So effect of human IRF1, truncated human IRF1 on the expression of apoptotic and anti-apoptotic protein in mammalian cell line. We have studied, and finally we conclude that C terminal of the IRF1 is required for the transcriptional activation of this protein. So without C terminal, this protein is not functionless. So here is the references, some references here, and I would like to acknowledge my PhD student. He got a scholarship during his research work, and also I am thankful to my head, Professor Rizwanul Haq, for providing the laboratory and infrastructure to carry out this work, and also the lab staff. Without them, the work would not be possible. So thank you all for patiently listening me. So I am happy if question will be asked for me.
So thank you very much, Dr. Krishna Prakash ji. Work has been done. This extensive work has been done in the recombinant DNA technology lab of the Department of Technology of the Central University of South Bihar. And the paper was on truncated mutant human cell line, and that was in search of an anti-cancer mutant protein. So it has been done extensively, and now the paper is open for discussion. The paper is open for discussion. Questions, if any.
Sir, मुझे एक सवाल I want to ask a question. I want to ask a question.
Yes, sir. Yes, sir. Please go ahead.
Is it audible to sir?
Yes, yes, sir.
Okay, so my question is that the truncation of the C terminal 82 amino acids you have removed, no?
92.
92, fine.
Yeah.
So is this has been done accidentally, or you have done sequentially of that?
No, we have done actually naturally it is also happening in some cancer cells. So cervical cancer, it is reported by Lee group BBRC that the isoforms of IRF1 is actually acting as a tumor progressor, not a anti-tumor activity. So that was the also purpose to delete this portion.
Okay, very fine, thank you. One more question.
Yes, sir.
And since my work is also on the truncation of a protein,
yes, sir.
I have done up to the 31 residues of a different protein.
Okay, sir.
So I want to ask just that what happens if any of the residues has been removed from the C terminal other than N terminal?
That is depending upon the protein, because my protein is having N-terminal DNA binding domain. So DNA binding activity we observed, but transcriptional activity we did not observe as transcriptional activity present at the C terminus of this molecule. In case of p53, the DNA binding domain is present in the central region of the protein. So that case, the activation domain is at N-terminal in p53. So that case, depending upon the protein, which domain is present, which side of the protein.
But sir, usually it has been seen that if amino acids are removed, the three-dimensional structures had been disrupted.
Yeah, you are right, sir. So, but in case my protein is modular protein, modular protein means each domain is function separately. If you remove one domain one by one, that domain functions separately to each other. So that's why we got the DNA binding activity. It's still DNA binding activity we got, but there is no transcriptional activity.
That's fine. That's very good. Thank you very much.
Thank you, sir. Thank you for the answer.
Thank you, Dr. Krishna Prakash ji. I
Yes,
had occasion to visit the BIT Misra campus of South Bihar.
Yes, yes. Actually, I was.
And meet the
I was. Yeah, I am actually just taken an exam, and university has pressurized me to evaluate the answer script and publish the result by 2nd August. So just imagine 40 students in batch, and yesterday exam was over, and 2nd August we have to publish the result. So maybe future, sir, I will visit you. I visit the physically there. Okay, sir.
I had an occasion to meet Dr. Rizwanul Haq Sahab there at the
Yes,
Patna.
sir. Yes. Sir is also from Darbhanga. The Dr. Ijanul Haq, Professor Nihal was the registrar of this university. So,
Now we have shifted to your own original campus at Gaya. So thank
yes,
you
sir.
for this very, very nice presentation.
Yes. Thank you, sir.
Thank you.
Thank you very much, sir.
Now I call upon Professor Santosh Kumar ji, retired professor and head of the Department of Botany and also Dean of the Faculty of Science, BRA Bihar University, Muzaffarpur. He was coordinator for 5 years of the UGC SAP program provided to this University Department of Botany, Bihar University, Muzaffarpur. Professor Kumar is a brilliant uh, exponent of the molecular biology stream, and we are really proud of him. He had published several research papers in reputed journals, and I request Professor Santosh Kumar to make his presentation on DNA methylation and PNA technology, that is peptide nucleic acid technology behind corona vaccine. That is the need of the hour. I think the entire world is doing its bit to bring out vaccines for the control of the ongoing corona vaccine, corona disease. So I request Professor Santosh Kumar ji to
Yeah, I am here. Thank you. Thank you.
within,
Thank
within,
you.
within the time frame of 30 minutes, sir.
Yes, thank you. Am I audible?
Yes.
And how to, Rohit, slide? Should I pick
Yes,
a window? Should I click
uh,
window?
sir. There are three dots just before the disconnection button, and there is a block and upside arrow in the bottom.
Yeah, upside arrow.
Yes, sir. If you will click over there, it will say that your entire screen, and you can share the minimized PPT by there only, sir.
Your entire screen has come.
Yes, sir.
And
then minimized PPT can be opened to share the screen, sir.
has it come?
We are just waiting, sir.
Now is it available on your screen?
No, sir. It's not showing right now.
Then
Uh,
share your entire screen.
yes, sir. You have to click it over there, sir.
Just a minute.
Yes, sir.
Has it come?
Yes, sir. Yes, sir. Yes, sir. Yes, sir. Yes.
Now I am audible
Yes.
Yes, sir.
and the screens are there.
Yes, sir.
Thank you, Dr. Vedanand. I am highly delighted to have been invited for in the August gathering, and I am highly obliged to the administration and the management for this symposium, especially Dr. Abhay Kumar and Dr. K.K. Sahu. I just retired from my post after serving the university, BRA Bihar University, for the 41 years. the inviting and giving a lecture for Darbhanga, for Mithila University, is always enjoyable for me, because not only I was born and brought, but my education, basic education from ML Academy, Lahariya Sarai. So this gives me a very good opportunity to speak before you, and working on the molecular biology out of the grant given by the UGC, which I could convert 2 laboratories in my universities, that is of molecular biology and plant tissue culture. I could able to go in sub-depth in these areas out of the UGC grant, as well as GST and a few other grants also. The present topic which I decided to speak before you is the demanding for the Corona-19 that is coming up in our day-to-day life, and this panic devastation of COVID-19, as we all know, that this has sickened and killed millions of people worldwide. The scientists are agog with the vaccines which have been tested in clinical trials to determine that they must be effective in stimulating our bodies to develop immunity and protect us from the disease. Therefore, COVID-19 vaccines are a critical tool in stopping the pandemic, resuming the normal life, and protecting ourselves and others from this disease. At the same time, I am surprised on the public, some of the public views over unfounded speculation that these vaccines could negatively affect our body system. At the moment, I want to remind that some 100 years ago, perhaps in 1918 to 1920, a Spanish flu, which came to even in our country too, costing 10 million lives. So reminding that a Spanish flu makes us a little bit embollished us, because COVID-19, which has crippled the world in terms of economy, human health, and life, I feel that if there is a loss of single life, it looks unbearable. But since efforts have been made, not in our country, but the whole of the world, so we are bound to appreciate the stakeholders to elicit safety by an effective vaccine for this novel coronavirus. I am sorry to present this particular slide because a few people are taking the advantages of social media. When somebody forwarded this slide, I was very much upset, because on the one way, we are trying to solve the problem, but on the other way, a few people are there, looks peddlers, they spread the fake news. If you go through this, you see it has advised watch till end, and also advised to share, which I never did. This lady, initially she is talking that persons are well after taking the vaccines, but she incited this person by adding fuel to the fire and asking what will happen in the future. This man, I don't know what is his name. He belongs to Canada. I feel that he is anti-humanity. His speech is completely anti-humanity because it is impossible for a messenger RNA vaccine to alter your DNA since synthetic messenger RNA operates only in cytoplasm and is incapable of entering into any cell, including nucleus. But this gentleman is making us in the absurd talk. His talk is nonsense, seems to be, and his talk is completely anti-human. I am sorry to say that he is one example that confirms difference between a pseudo doctor and a scientist. What he says is rubbish. How the people can afford to forward such nonsense talk? Such approach should be punishable. Returning back to my topic of COVID-19, we all know that a virus causing severe acute respiratory syndrome, otherwise known as SARS-CoV-2. This is due to a strain of coronavirus not previously seen in human. Coronaviruses, as the name clicked, because it causes crown-like spikes on their surface. It is a large family of viruses circulate in humans as well as in some animals like pigs, bats, and cats. We all know that first time it is reported by in Wuhan, China in December 2019. And following the epidemic, pandemic was recognized by World Health Organization in March 2020. This I have presented an animated visual of coronavirus, two-dimensional. You see the genetic material. Since viruses contain either DNA or RNA, this virus contains RNA enclosed within the membrane, and on this surface of this membrane, they have spike proteins. Like the other members of this viral family, SARS-CoV-2, which is the official name for the coronavirus, it is an RNA, as I told you. Each individual virus is composed of single strands of genetic material. We remember HIV virus also is RNA virus, but there are two copies in that. Here it is a single strand covered by a fatty layer coated with a spike. These spikes are what the viruses uses to hijack our cells and use our molecular machinery to make more copies of its. SARS-CoV-2 spreads easily in persons. We have been victim of this in our country in the first and second phase. It goes through respiratory droplets or by touching any contaminated surfaces. Some people who are infected might not exhibit symptoms. You know, we all know the symptoms: the fever, cough, as well as some problems in neck. But the persons who may not exhibit symptoms can spread the virus to other people. COVID-19 has been found to be serious for people having comorbidities, who are suffering from heart or lung diseases and diabetes. It is reported that 65% individuals with comorbidities diagnoses SARS-CoV-2 infection. Symptoms can start minimum 2 days up to 14 days after exposure. After exposure, it takes 5 days to have taking the average time. People with COVID-19 symptoms can slowly get worse. They become ill after a week once the symptom starts. Since it is related to immune system, I will like to introduce some of the WBC which are involved in fighting the infection for any viruses. They are macrophages. It swallows and digests germs. They can leave a part of the invading germs, and that are called antigens. The B lymphocytes, we are familiar with these producing antibodies, and T lymphocytes, which attack cells by producing lymphokines of different types and induces B lymphocytes to produce antibodies. As we all know, the first time a person is infected with the virus that causes COVID-19, it can take several days or weeks for their body to make and use all the germ-fighting tools needed to get over the infection. After the infection, the person's immune system remembers what it learned about how to protect the body against the disease. The body keeps a few memory cells of T lymphocytes as well as B lymphocytes. Though they go into action quickly if the body encounters the same virus again. Thus, lymphocytes, after producing antibodies, attack them. However, the experts are still learning how long these memory cells protect a person against the virus. There are different ways to make a vaccine. Coming in order to solve the problem. But the ultimate goal of any sort is to introduce a biological equivalent into the body so that if the real enemy enters anytime, our immune system knows how to fight it off. There are several different types of vaccines. Each type is designed to teach one's immune system how to fight off certain kinds of germs and serious diseases that they cause. But for creating vaccines, there are some considerations. The considerations are how your immune system responds to the germ, who needs to be vaccinated against the germ, and the best technology or approach to create the vaccine. We all know that COVID-19 vaccines help our bodies to develop immunity to the virus that causes COVID-19. The body is left with a supply of memory T lymphocytes as well as B lymphocytes, as I told you. And that helps in remembering our system to fight the virus in the future after infection. It takes a few weeks. This is an important point to learn, that after vaccination, a few weeks are involved to produce T and B lymphocytes. So it is possible that if a person who could be infected with the virus that causes COVID-19 just before or after vaccination and then gets sick because the vaccine did not have enough time to provide protection. Sometimes after vaccination, the process of building immunity can cause symptoms. Again, those symptoms which are normal and not coming in way of any casualty. This is the preliminary ideas which everybody knows, that the vaccines have been developed for the last 50 years or more than that. They can be categorized as inactivated virus, as in case of hepatitis, flu, and polio. Live attenuated vaccines, attenuated, matured enough, as in rotavirus, MMR, mumps, smallpox, chickenpox. There are few vaccines also which are recombinant polysaccharides and conjugate vaccines, which are used in hepatitis B, whooping cough, meningococcal and pneumococcal diseases, toxoid virus. But we are concerned here, since for the COVID-19, we have used messenger RNA vaccines as well as viral vector vaccines. Concentrating on COVID-19 vaccines, what have been used throughout the world, they are either messenger RNA vaccines that contain part of RNA sequence of coronavirus that specifically produces spike proteins. Protein subunit vaccines include harmless pieces of protein of coronavirus, and then comes vector vaccines. Inside the cell of the modified virus, there is the genetic material of coronavirus that has been engineered in the vector RNA, vector nucleic acid, that produces protein. This is called viral vector. And all these produced proteins behave as antigens. Taking one example, how viral vector vaccines are prepared, they are a modified version of different virus as a vector. Adenovirus has been taken as a vector to carry the part of the genetic material of coronavirus. This adenovirus, which causes common cold, has been used as vectors. Adenovirus is one of the viral vectors being studied in clinical trials. Scientists used this technology to make COVID-19 vaccines as well. Concentrating how the Covishield vaccines have been prepared, that Covishield vaccine contains a non-replicating virus, that is weakened chimpanzee adenovirus that causes common cold. Since viruses are specific, we are not affected with the adenovirus. That virus has been genetically engineered to produce SARS-CoV-2 spike glycoprotein in the body. But the virus is weakened and cannot cause any disease. It provides active immunization against COVID-19 infection that helps us to fight the virus if exposed in the future. That means there is a reduced risk of developing illness and further consequences. How viral vector vaccine works? Initially, the vector enters a body through the vaccine. It enters into our body to produce a harmless piece, a spike protein of coronavirus. Then the body cell displays the spike protein on its surface, and our immune system recognizes it as an enemy. This triggers our immune system to begin producing antibodies and activating other immune cells to fight off what it thinks is an infection. At the end of the process, get this protection from the vaccine, any temporary discomfort, as I narrated, the symptoms, which is partially an indication that the vaccine is working. But it is sincerely, it has been found that they never affect our DNA in any way. Coming to the Covishield vaccine virus has been developed by AstraZeneca with Oxford University in the United Kingdom and is being manufactured in India by the Serum Institute at Pune. Successful clinical trials have been conducted in South Africa, Brazil, and the United Kingdom, with bridging study results in the Indian population. Based on these facts and figures, our Drugs Controller General of India has approved. Coming to the messenger RNA vaccine, COVID-19 messenger RNA vaccine give instructions to our cells to make a harmless piece, as I told you earlier. And what is called that protein is a spike protein. The spike protein is found on the surface of the virus that causes COVID-19. How it works? First, immune cells use COVID-19 messenger RNA to make protein piece. After the protein piece is made, the cell breaks down the messenger RNA and gets rid of it. Next, the cell displays the protein piece on its surface. Our immune system recognizes the foreign protein and begin building an immune response by making antibodies. Messenger RNA, you must recall, never enters the nucleus of the cell, which is where our genetic material is kept. At the end, our system learns how to protect against future infection. Here, during the preparation of the vaccine, a few problems have come up in the form of barrier. We noticed that inflammatory effect on the body as well as fragile nature of the molecule. But two scientists, Westman and Carrico, in the United States have covered, and by utilizing messenger RNA technology, they could able to improve the two barriers, these two barriers. The messenger RNA vaccines in use today are the first of their kind. However, amid a devastating pandemic, has this technology reached the mainstream prominence? How messenger RNA vaccine works? The RNA-based genetic sequence of the coronavirus was first turned into DNA by reverse transcription, as we all know, which allowed them to identify the instructions necessary to create the spike protein. Then engineering was done, and the synthetic messenger RNA that was packed into the vaccines in the form of vaccines. Once it is ribosomes interpret the message coded in messenger RNA to make proteins and start executing its instructions, for which the Philip Sharp in 1993 got the Nobel Prize for his contribution and understanding. One of the lymphocytes that has been recognized as watchdogs of the immune system, dendritic cells. They play an essential role in responding to pathogens. They patrol the body in search of foreign invaders, and when they find one, they start stimulating an immune response by clicking to T cells in order to form antibody from B cells. When these cells encounter messenger RNA that has been injected via vaccination, their ribosomes decode the message and allow the cells to temporarily display spike proteins. In the Nature, in June 2021, I found a paper, adenovirus can train the body to continue to make more vital T cells even for a long time once antibodies are formed from the vaccine. As per the study, the vaccine not just generates antibodies against the SARS virus, also created training camp in the body to enable search and destroy even a new variants. This is most interesting paper which I saw in Nature. Actually, what has happened recently, it has been made very clear that there is a special gene in our nose. Every cells have the different genes, but it is functional in our nose, which plays a main role in attracting this deadly virus. This gene is producing a protein, an enzyme, ACE2, angiotensin converting enzyme. This gene present in the nose is the first sensitive site where the virus first attacks and enters human body through ACE receptor enzyme site. This is the enzyme site, if you can find, which receives the virus, which welcomes the virus. This is the biggest reason for infiltration of coronavirus. This ACE2 gene present in the nasal epithelium is highly reduced in children. However, it increases with age in the human body. It gives a knowledge at this particular time that children, why children are less prone to this disease, because ACE2 gene is in reduced form and it increases with the advancement with the age. It is also found that this particular protein is more prevalent in men than in women. How to corner this virus? Two ways I have adopted. One is methylation and other is DNA. We all know that DNA methylation occurs in cytosine nucleotides by DNA methyltransferase at position, fifth carbon position, hence its name. Actually, methylated cytosine occurs in pairs, in islands, CG islands. This 80 to 90% CG islands and remaining 10 to 20% remained unmethylated. In the promoter of any gene, CG methylation reported to facilitate. If there are less number, it is hypomethylation. If there are good number, then there is hypermethylation. Accordingly, the high expression of this ACE gene in the lungs, liver, and brain lies within the sparsely methylated CG islands.
Please conclude the address.
Okay, coming to the final point, cysteine proteases that reduces hypomethylation covers, and how to recognize the methylated structures. For that use, we use isogizomers, which cut the methylation never allows any enzyme to prevail. But isogizomers, there are three. One example I have taken, MBO, SAU3, and DPN, which can digest as per, if the digestion is complete by MBO, if the digestion is partial by SAU3, and if there no digestion in case of DPN, this reveals that only cytosine is methylated, not a single adenine is methylated. Coming to the next part, PNA. The US Food and Drug Administration has recently allowed a company at Republic of Korea for emergency use of COVID-19 RT-PCR peptide nucleic acid kit recently on May 24, 2021. This SARS-CoV-2 nucleic acid is first extracted, isolated, purified, and that put into the PCR amplification, where the detection of hybridized fluorescent peptide nucleic acid groups has been utilized. This peptide nucleic acid for the first time in 1991 was synthesized by Peter Nelson and Burkhardt from the University of Copenhagen. PNA is not known to occur naturally. So it is a hypothesis that maybe it is similar to DNA and RNA, but differing in the composition of its backbone. PNA mimics DNA, but instead of the glycosidic bond, they do have the amino ethyl glycine linked with the peptide bonds. This is the one side, one chain of nucleotide in the PNA. You clearly see the difference between the PNA and DNA. PNA can be attached with the hydrogen bonds. These are the different oligomers, adenine, cytosine, guanine, and thymine oligomers of the PNA. PNA is capable of recognizing a specific sequence of DNA or RNA by following the Watson-Crick hydrogen bonds. PNA is capable of inhibiting transcription. So it qualifies for investigation of coronavirus, its detection. Because of the thermal properties of DNA, you see, if you attach PNA and DNA, the TM value goes to 69%, which is highly used in polymerization in the DNA amplification. So PNA and RNA also to have high degree of thermal stability, that is 72%. The PNA have other functions, a stronger binding independent, salt, greater specificity, resistance to nucleases, and insolubility. So finally, I like to conclude that all these properties speak for PNA, which can replace DNA as a probe for many investigations, including COVID-19. Further research is warranted to validate the authenticity of the recent discoveries just discussed. So with these words, I was completely thinking in terms of methylation and the PNA to be another feat if being pursued, and it has to be proven clinically by the doctors whether they amplify, whether they are good resources, if the vaccines can be controlled. With this, I think that I have presented my viewpoints on the topic which I have just discussed. Thank you. Thank you, Dr. Bhavnan.
Thank you, sir. Thank you very much for the wonderful discourse that you have presented regarding the thinking, regarding that is going on, the scenario that is being discussed all over the world regarding the preparation of vaccines for the control of COVID-19. There is one question for you, sir.
Yeah.
What the question is from one Ananda Prakash that is there in the chat box. What is your view upon the efficacy of vaccine on the mutated strain of COVID-19 during second wave? Because the vaccine was prepared in 2020 for the first strain which came in 2020, that is the same year.
No, the efficacy has been clearly defined that different vaccines like the Covishield, it goes to 71%, and the Moderna and Pfizer vaccines they have claimed to up to 91%. So there is no question. I myself have taken both doses in April, and I am feeling well and fine. So the percentage can vary. The scientists are working day and night to improve the situation. And if recently the PNA kit, which I have just introduced, is likely to increase the values which they claim. This is a Korean company, South Korean company, which is doing, and United States of America, the Food and Department has agreed to give this, permission.
There is one question from my side also, sir.
Yeah.
The mechanism behind the production of nasal vaccine.
Nasal. Yeah, that gene has been identified in the nose. And as I told you, why you take the example, children are taking less susceptible for this disease. Because of this reason, that the gene is not active in children, it increases its activity with the passage of time when the age increases. So nasal vaccine will be another our victory if the particular nasal vaccine comes, which I think next month or 2 months it will be introduced to the general public.
Thank you, sir. If there are any more questions from the audience. So thank you, sir. Thank you for the wonderful presentation. Thank you, sir.
Okay, thank you, sir. Thank you.
Okay, sir. And now the second technical session is going to be started, and the chairperson will be Professor T.N. Jha, of the retired professor of this department, LNMU University Department of Botany, and the reporter will be Dr. Khalid Anwar. So the next technical session will be started. Professor Jha, please share. Yes, sir.
I'm happy to introduce to you next speaker, Dr. K. Marsan. He has been the lecturer professor and vice chairman of IQAC Banaras University. He has also been dean and head of Institute of Agricultural Science, BSU. He has several years of teaching and research experience. Dr. Marsan published more than 50 research papers in respected journals. His area of expertise is microbial genetics. So, Dr. Parsampine, you are requested to deliver your lecture.
Thank you. Thank you, the chairman and Professor Abhay, Professor Sahu. Is it that my presentation is coming? Can you see my presentation?
No, sir.
But I have shared it. It has already been shared. Can you see it? The
Yes,
title?
Yes, sir. Now it is visible, sir.
Yes, is it visible?
Yes.
Yes, sir.
Can you see the title?
Yes, sir. Yes, sir.
So, good morning, ladies and gentlemen. Well, I feel so happy to be here today among you all on this web platform to talk on molecular genetics of nitrogen fixation and its regulation in desert a boon to human life. That's certainly a subject of paramount importance to keep us at par with the rapidly advancing world of knowledge jewels in the national global scenario on the subject, its perspectives and strategies, overarching the goals of harnessing the unique properties of useful microbes with a higher spatial resolution within the live green energy application sector, blended with a soft touch of fundamental science to trigger exceptionally well-planned, sophisticated researches on utilizing some of such gifted bacteria and cyanobacteria, which occupy a major proportion among the microbial diversity. I know microbial diversity is the biggest tool to interdisciplinary life sciences, whether it is health, hygiene, nutrition, physiology, biophysics, biochemistry, food science, toxicology, environment, agriculture, genetic engineering and biotechnology, or even cosmetic technology, fermentation technology, and allied industrial sectors. And these microbes were the first colonizers on the earth 4.5 billion of years ago. Earth separated from the sun as a lump of fire. After it cooled down nearly 3.2 billion years ago, the two components inherited from the sun, the inanimate and animate, came into existence, came into expression. The inanimate transformed into solid stones, which later formed the sand and then to soil and agricultural soil, whereas the poisonous liquids converted gradually to water and then to drinking water, and so were inert gases that were converted to poisonous gases, and now the useful gases like oxygen, hydrogen, nitrogen, etc. On the other face of the coin, the animate components reassembled in the form of what we know today as DNA and RNA, from where bacteria and cyanobacteria originated. With metamorphosis, plants and animals came into existence up to human beings. And under the death and decay of these plants, animals, and human beings, they were compressed in between the stone particles, and hence fossils were created and the same fossil fuel we find in the form of coal, petroleum, and then the power or electricity which creates this present world where we are present. Now, the last month, Nature published the paper that diamino phosphate was present on the Earth to support knitting of formation of DNA backbone right 3.2 billion of years ago, and these were present in the first bacteria and cyanobacteria. Now, if we leave aside many of the microbes and concentrate upon only the plant growth promoting rhizobacteria, I mean the PGPR, these include all natural fixes, whether symbiotic, asymbiotic, or free living, most of the phosphate solubilizers, most of the growth hormone producers, most of the siderophore producers, and most of the biocontrol chemical producers. And by further narrowing down only to nitrogen fixes, nitrogen fixation is catalyzed for the entire biosphere by virtue of the enzyme nitrogenase. The enzyme nitrogenase is highly conserved by dint of the evolution and continuity of NIF genes, coding for its both components. As many as 21 to 23 NIF genes may be there under 8 to 9 transcription units, of which NIFH is highly conserved, a molecular cloning and sequencing of which has produced a large, rapidly expanding database of sequences from the diverse aquatic and terrestrial flora, which I shall be describing further. Now, we see a paradox in the atmosphere, in nature, in the sense that we have only 0.03% of carbon dioxide, but all green plants and those microbes carrying chlorophyll A have the capacity to absorb this carbon dioxide by virtue of the enzyme ribosco present there, and they manufacture their own food. Whereas, in contrast, the atmosphere has more than 78% of nitrogen gas, but no plant has the gene, the genetic machinery, there's a NIF gene, so that nitrogenase enzyme can be prepared because it's the only enzyme that can break the very, very strong triple bond between two N atoms. And that's why this nitrogen gas cannot be directly utilized by the plants. And hence, we require some bacteria and cyanobacteria which carry NIF genes and produce nitrogenase enzymes. So the only alternative which remains under the genetically determined discriminate use of CO2 and N2 by plants is to have a prokaryotic NIF genetic engineering of most of the cereals, pulses, pulses, vegetables, fruits, and flowers. That's the only answer at this juncture. So far as synthetic nitrogen fertilizers are concerned, through Haber and Bosch process, by providing 450 degrees Celsius of temperature, the two N atoms are separated, the triple N bond is broken, and then it is joined with hydrogen to produce ammonia or urea fertilizer in the factories. So it consumes a lot of non-renewable fossil fuel reserve that is spent in creating such high temperature, and then transporting this synthetic fertilizer in the form of urea, ammonia to the agricultural fields. Moreover, through radioisotopic studies, it has been confirmed that 75% of this synthetic fertilizers is diverted to plants' robust vegetative growth, and only 25% goes to grain filling, because these days we use high-yielding varieties, and due to high fertilizer requirement, these become a giant body. So in the same proportion, they require all other compounds. Hardly we give N, P, and K. Some progressive farmers give some zinc, some boron spray, etc. But what about the other elements? The plants require 103 elements from the soil. So these 103 elements, I mean 96 to 97, are consumed mercilessly, squeezed out of the soil, and the soil becomes sterile by the use of high-yielding varieties with the help of synthetic nitrogenous fertilizers. On the other hand, if you use biological nitrogen fixers, these require nothing but only solar energy and water to produce ATP, and the nitrogenase is capable enough to break the strong N bonds between two N atoms of a nitrogen molecule and to join it with hydrogen to form ammonium ion or ammonia. This is self-renewable in cultivable agricultural lands, most economic for biotechnological production, distribution, and use. And the radioisotopic studies have shown that 90% of fixed nitrogen from the biofertilizers from cyanobacteria, bacteria contribute to grain fertility, and only 10% go for the vegetative growth of the plants. So plants may be smaller in size, but may be a beautiful quality material. And these also secrete growth hormones, biocontrol agents, and replenish soil by micro and macronutrients from the soil already exhausted. So these help restore, maintain, and enhance soil fertility. The highly efficient nitrogen strains available for each crop, means crop specific, are now being cultured and supplied to the farmers. Now, if you see the table, industrial fixation annually is 60 million tons through Haber-Bosch process by creating 450 degrees Celsius of temperature, utilizing our fossil fuel. Through lightning, it is 5 million tons. Means through lightning, lot of temperature is created in the sky, and the nitrogen gas is broken into N atoms. Similarly, the rainwater is broken into protons and electrons. N and H are joined. So the concept that the rainwater is distilled water, it is not so. It comprises a lot of fixed ammonia inside the upper sky. Through human animal waste, we get 10 million tons. Through green manuring, 15 million tons. But still, the biological nitrogen fixation is 100 million tons per hectare per year. But the lack of cooperation, coordination between agronomists and microbiologists has created a problem so far that we are not going to consume the entire capacity of our earth for the biological nitrogen fixation in agriculture. The specialty of nitrogen fixation is that it is not operative at all in any eukaryote. It is operative strictly in some of the prokaryotic microorganisms like some of the bacteria and cyanobacteria, either asymbiotically or symbiotically. It's a highly anoxygenic biological process. Both NIF gene and nitrogenase enzyme are extremely oxyphobic, switched off or destroyed irreversibly under excess oxygen. And both NIF gene and nitrogenase enzyme complex work only under strict ammonium conditions. Means if already fixed nitrogen is there in the form of ammonia or urea, NIF gene cannot be switched on. Now, which are the organisms? The first is strict anaerobe, which fixes 18 to 30 kilogram nitrogen per hectare per year. Why it is in archaebacteria? Because it is the same bacterium present from the time of the origin of the earth. The proof for which is that it is present in the hot springs of Kullu, Manali, Rajgir, etc., even in the volcanoes of Japan above 100 degrees Celsius. And in these bacteria, NIF genes are present. So NIF gene is a gene of oldest origin, right from the time of the origin of the earth, origin of life on the earth. And eubacteria like Bacillus polymixa, etc. These are the same bacteria with the same quality, but present on the ambient right now temperature of the earth. And this report has been published this morning that we have got 3.42 billion years ago fossil record of these archaebacteria in which NIF genes were present. The same components have been chemically analyzed by HPLC and found to be similar to the archaebacteria present in the Kullu, Manali, and Rajgir hot spring and volcanoes of Japan. So you can see how old is this NIF gene. The matter is that we have to scientifically utilize it in the fullest manner so that it may be of use to our agriculture. The next category is facultative anaerobes, which fix 18 to 33 kg of nitrogen per hectare per year. These are beautiful microbes like Klebsiella pneumoniae. It is supposed to be the model nitrogen fixing bacterium because all components of NIF gene are present together in close proximity in this bacterium. But the problem is that this is aerobic bacteria, but with the special gift from nature that whenever it undergoes a nitrogen crisis, it adopts anaerobic mode and then nitrogen, the NIF gene is induced, and lot of nitrogen is synthesized and nitrogen is fixed. So our Enterobacter Escherichia coli is a non-nitrogen bacteria, but we introduced NIF gene through genetic engineering. This strain is preserved in the American Type Culture Collection Center, Rockville, near the University of Maryland. Now, next is microaerobes. Microaerobes, there's no problem because uh, it is aerobic, but the threshold level of oxygen is below the damaging level of nitrogenous or nitrogen, like Aquaspirillum, Azospirillum, Arthrobacter. These are the examples. These also fix 18 to 33 kg of nitrogen per hectare per year. The other category is pure aerobes, like Azotobacter, Bergeringia, Dirksia. How oxygen is not damaging the NIF gene, how NIF gene is protected. It's a beautiful story. I will tell you in the later slides. And then the non-oxygen evolving autotrophs. Autotrophs means like sulfur bacteria, iron bacteria, Rhodospirillum, Rhodospirillum, etc. There's no problem because being photosynthetic, it is not oxygen producing, so nitrogen is safe there. But the most problematic is pure aerobes like cyanobacteria or blue-green algae, where photosynthesis is similar to that of higher plants. It's a typical oxygen evolving photosynthesis, or better to say the oxygenic photosynthesis. How nitrogen is protected, I will tell you. Then comes the associative nitrogen fixes, 27 to 45 kg nitrogen per hectare per year, like all the previous organisms. And these are Azospirillum brasiliensis, Pseudomonas, Enterobacter, Bacillus, Klebsiella, which fix nitrogen in close proximity to the root hairs of cereals like rice, maize, and wheat. They have been found. And they are being exploited for the agriculture use beautifully. Now, the most important is the rhizobial group, legume rhizobium symbiosis, you must have heard. Now, rhizobium has been renamed as Cynorhizobium, Bradyrhizobium, Mesorhizobium, Allorhizobium, and Azorhizobium. Cynorhizobium are those which infect the peculiar pulse plants like pea, alfalfa, trifolium, fasciolus, soybean. But the species of Cynorhizobium is different, like Monstera, Melutite, Trifly, etc. Because very specific rhizobium only can infect for nodule formation in a specific legume. I will tell you later in the next slide. And bradyrhizobium are those which are slow-going rhizobia. They infect Vigna and Cicer. These have the property that this can grow ex planta condition also under nitrogen-free medium. So most of the genetic studies on rhizobial NIF genes have been made with this bradyrhizobium because other rhizobium cannot grow in nitrogen-free medium. You will have to supplement with nitrate, nitrite, or ammonia, and then its NIF gene will be suppressed. So bradyrhizobium is a beautiful material for genetic and molecular genetic studies. Mesorhizobium is the one which infects lotus. Allorhizobium infects Parasponia, Neptunia. And Azorhizobium is the only rhizobia with only one species, Colinodons, which infects Sesbania rostrata. That's the only plant, Sesbania rostrata, prevalent in Tamil Nadu area. It has both stem and root nodulation. That's why the rate of nitrogen fixation, here it is 300 kg, there it is up to 100 to 600 kg nitrogen per hectare per year. So it's a very great material, Azorhizobium Colinodons, but the problem is that it cannot infect any other pulse crop. Now, what is the specialty? The pulse plants in their root hairs produce lectin of various serological qualities, various categories. And the rhizobia produce ricadhesin, CA2 binding protein. And unless this ricadhesin and lectin match each other, these cannot infect. Like a lady purchasing a red sari from a shop and then moving for a blouse piece of matching color to the blouse piece shop, the shopkeeper will find the match out of 100 shades of red and will stop himself at one place where the exact match is there. So unless there's a exact match, this ricardicin cannot infect a particular lectin. Means that's why Lemnocerum, Rhizobium lemnocerum infects P only, not the fasciolus. Rhizobium fascioli infects the fasciolus, not the P. And thing is with corona, Professor Santosh Kumar were dealing just now. It's a beta corona which infects human beings only. So beta corona is in search of human beings. It's not alpha, delta, or gamma corona that infects the other animals at Tigris in New York Zoo or the swine flu or many other birds and other things. So beta corona is specific. That's why we have to protect ourselves either with vaccine or even after vaccine with the help of mask, etc. So the same thing is here. Legume rhizobium symbiosis, it is very, very strain specific due to serological homology. Now we have some non-legumes also infected by some of the bacteria like Frankia, actinomycete, Elder, Casuarina, Hypophy. These are non-leguminous plants where nodule is formed by the action. And these are also very good nitrogen fixer, but not much, 50 to 75 kg, but the quality is very good. And then coming to the other bacteria and cyanobacteria, Azotobacter pastelii and Spirillum notatum are the bacteria which infect the maize and sorghum, digitaria. They infect the root sheath inside, outside epidermal layers and fix a lot of nitrogen, 35 to 40 kg of nitrogen per hectare per year. And cyanobacteria is capable of making this association with almost all groups. Lichens means with fungus in the mycelial mesh. Anthocerus liverworts, nosto cavities are there. Azolla inavena is a well-known in the dorsal leaf cavity. It fixes remarkably 300 to 600 kg of nitrogen per hectare per year. Macrozomia means gymnosperms, Nostogonovina, coralloid root we find. In Gunnera angiospermic plants also, nostoc is present, leaf vesicles. And in Azolla complex, we worked on this. It has a problem in North India. It is highly thermosensitive. It's highly P requiring, and its spores do not really germinate. The insect pest infestation is also very high. So we got a mutation to MNNG, N-methylene nitro and nitrosoguanidine. And then we found this paper published in Nature and Oxford that we found mutants which are now 50% low phosphate requiring. Mutants which can tolerate high levels of nitrogen up to 40 degrees Celsius, and the mutants that can avoid the insect pest infestation, and the mutant that can have readily spores. You can take a handful of spores, throw it to the rice fields, waterlow condition, and to germinate into us plenty of azolla. So there will be no problem of transportation of azolla. This paper very much appreciated. But in North India, there's a problem that the temperature goes even beyond 40 degrees Celsius in the rice growing seasons. But these strains are working well in NRRI, Cuttack region and TNAU, Coimbatore region. Now coming to the cyanobacteria, I was telling how NIF gene is protected. Whenever there's nitrogen crisis, the het gene is triggered to form heterocyst after every 19-20 cells. It's a metamorphosed, bigger hyaline structure, double-layered, thick wall, outer wall of polysaccharide, and inner wall of glycolipids. It complete protection from external oxygen because of the thick wall. And internally, photosystem second is removed out of genetic rearrangement. I will come on the gene and then tell you how. So there's no oxygen involved and no photosynthesis, no oxygen evolved. So that's why heterocysts and vegetative cells maintain a kind of symbiotic relationship in that the heterocysts fix nitrogen and supply to the nearby vegetative cells. And in return, the vegetative cells provide with carbon selectin and reductants as well as ATP for supply, although photosystem 1 is present in heterocysts and enough ATP is produced. In Oscillatoria, the non-heterosister forms, so far we knew that the Oscillatoria is non-nitrogen fixing, but it's not. The recent researchers have shown that these two have NIF genes. These produce nitrogenous enzymes. But the problem is that there's nothing like heterosis, but they have a TS gene, temporal separation gene. Whenever there is a this temporal separation gene is triggered, which stops photosynthesis for a while. And no sooner photosynthesis is stopped, oxygen evolution is stopped, NIF gene is triggered, it expresses, synthesizes nitrogen in enough quantity. Nitrogen is filled with the fixed nitrogen, and again TS gene is back to place, and again it becomes photosynthetic. So this is the unique information. So none of the cyanobacteria are now non-nitrogen fixing. They're all nitrogen fixing, but only the condition that TS gene has to be triggered. Then we have some microorbic bacteria I was talking, Azospirillum, Aquaspirillum. These are present on the thick mucilus ball, which protect from external oxygen to invade. And internally, the rate of oxygen evolution is so low that it is within the threshold level of damaging NIF gene or nitrogen enzyme. So NIF gene is protected here. The more interesting is Astrobacter situation, where it is highly oxygenic, but oxygen is not injurious to NIF gene as such. It gets dissolved in the aqueous medium of the cell and converts to superoxide radicals. But then Astrobacter has a specific gene which is a constitutive gene for the production of enough production of superoxide dismutase and catalase, known as Fe-Sethna protein, you must have read. And this has the capacity to dissociate the superoxide radicals and convert it to hydroxy and peroxy free radicals, which are made volatile. And that's why NIF gene is secured here and lot of nitrogen fixed inside the Astrobacter system. It's a very powerful organism these days to be used with wheat and rice. And coming to the leg rhizobium symbiosis, in the root nodule is there, but then there are involvement of various genes. Even the plants have 3 genes, known as noduline genes. The 3 genes out of which first is responsible for forming nodule structure, second for internodular enzymes, third for globin protein, that is leg hemoglobin, which acts like oxygen buffer between oxidize Fe3 to reduce Fe2 forms, keeping bound oxygen and free radicals level in the ratio 10,000 to 1. And this leg hemoglobin pigment, which keeps on moving from cell to cell, the entire nodule is partitioned to various chambers. And if leg hemoglobin is present in one chamber, it will consume the entire oxygen for a while, like pranayam of Ramadev Baba. And that's why that particular chamber will be oxygen free. The bacteroid will fix lot of nitrogen, and then the leg hemoglobin will move to the other chamber. So this way, nodule doesn't fix nitrogen the whole as a whole for all the time. It fixes nitrogen partially from chamber to chamber, chamber to chamber. And in addition, the rhizobium has 15 NOD genes for root colonization, root adhesion, rare branching, hair deformation, host specificity, etc., which I have already summarized in my earlier slides. So by the combination of both bacterial and plant genes, a beautiful nitrogen fixation, a very, very powerful nitrogen fixation is there in legume rhizobium symbiosis. Now coming to the biochemistry, the time is very short. Some of my time was snatched by Professor Santosh Kumar, but no problem. I would like to finish it up in time. You see, ammonia is the key point, ammonia man. The cyanobacteria or bacteria carrying the NIF gene are blessed with the property to produce nitrogen enzyme, which consumes nitrogen, breaks its triple bond and reduces it to ammonia man, utilizing some ATP, converting to ADP. In plants and animals, there's nothing like nitrogenase or NIF gene, but these can reduce nitrate by the use of NRD gene present, which produces nitrate reductase enzyme, converts it to nitrite. And then the plants also have nitrite reductase gene, which that is known as NIT gene, produces nitrite reductase, converts nitrite into ammonia. So ammonia is the joining point. Whether ammonium received by the nitrite fertilizer or directly as ammonium fertilizer, or it is given by the biofertilizer bacteria or cyanobacteria in the form of converting the elemental nitrogen into ammonia. So now ammonium is the starting point, whereafter four primary aminating enzymes are there. First is GS gene, which produces glutamine synthetase, and the first amino acid formed is glutamine. Second gene is Bogart gene, which produces glutamate synthase, and it converts glutamine to glutamate. Third gene is GDH, which produces glutamate dehydrogenase, which converts glutamate to alanine. And the fourth one is ADH gene, which produces alanine dehydrogenase to convert alanine into other amino acids. And that's how the entire amino acids are formed and proteins are synthesized. Now, coming to the nitrogenous biochemistry, it has two components. The bigger one is 240,000 molecular weight with four chambers of 60,008. The chemical constitution is 2 molybdenum 26 to 8, iron and 28 to 30 sulfur components. And the second component is known as nitrogenase reductase. The component first was primary nitrogenase. Second one is nitrogenase reductase, which has two chambers, mirror copy, known as gamma component. It has only 60,000 molecular weight. It is composed of 4 iron, 4 sulfur components. So starting from component 1, it has the activity to absorb the ferredoxin or flavodoxin generated at many points in photosynthesis respiration. And these ferredoxin or any other reductant, FADS2, etc., are broken into protons and electrons. And these protons are subtract to be joined with nitrogen in beta chamber. Alpha and beta are two chamber of component 1 in mirror copies, 2 copies. In alpha chamber, there is the FeMO cofactor of 5,000 molecular weight, comprising of 1 molybdenum, 8 iron, 6 sulfur. This is the site where elemental nitrogen for atmosphere is absorbed and broken into N ions. And from beta chamber, protons are received and N and H are joined to release NH3. So this is the machinery. And this is in 2 copy because it has to be continuous process like the carburetor of an engine. So long as the alpha 1 is busy in breaking the triple bond of nitrogen, the alpha 2 has already broken, it is busy in joining the N and H bond to release. So it's a continuous process. So proton storage is also in duplicate. The nitrogen fixing component is also in duplicate, and the proton supply units are also in duplicate. So MO has a key factor here, key role in nitrogen fixation. But we have some organisms like known as V nitrogenase organisms, where the work of molybdenum is taken over of vanadium in Clostridium. Still, we have some recent Clostridium species in which neither molybdenum nor vanadium is there. It's known as zero nitrogenase. And lot of ATP is also consumed, ranging from 4 to 29 molecule of ATP for releasing 1 molecule of ammonium ion. So it varies. In some bacteria it is 12, and as bacteria 4, in Klebsiella it is 29. So this way the natural process goes on. And this diagrammatic structure, 1 molybdenum, 8 iron, 6 sulfur units comprising of this iron molybdenum cofactor. This cofactor has been taken out and put to the test tube. If you supply some hydrogen source like Na borohydrate, even in the test tube nitrogen can be fixed. These are the recent discoveries in the diazotrophs. Now there is another gene known as Hoop gene, that is uptake hydrogenase gene. What happens is sometimes, see, this is the component 1, sometimes in your mobile, sometimes if there is some impurity in the carburetor, your scooter, motorcycle will be stopped. You will have to clean the carburetor. So similarly, what happens is sometimes there is some time taken in breaking the triple bond of 2 N atoms in the alpha subunit. By that time, the proton coming from beta 7, it cannot wait much and it escapes in the sky. So if the organism is provided with a hook plus gene, uptake hydrogenase enzyme is there, it will capture that escaping proton and recycle it back to the beta 7. So it will be most economic. Otherwise, it will be a loss because consume a lot of ATP. So hook plus strains are much more efficient nitrogen fixers than the hook minus strains. And this is the diagrammatic structure of the NIF genes. It has the genes are present in two tiers. On the lower tier, 23 NIF genes are present in 24 kilobase pair sequence. And on the upper tier, there are four genes. These are primary immunity enzyme genes, which I discussed earlier, the GS, GOGRT, GDH, and ADH. These are known as GLN genes in case of cyanobacteria, NTR genes in the case of Azotobacter and Klebsiella, and fixed genes in the case of Rhizobium. These are present under 9 transcriptional units, and this is the detail. You see, there are a lot of this genetic background is present in Klebsiella pneumoniae. In all other natural fixers, you will find the some part of NIF gene at some place, some other part in some other place. But it's only in Klebsiella pneumoniae that all genes are present at one place in continuous unit, just adjacent to the histidine producing gene. So these are present in 9 transcription units. You see the QB, AL, FM, ZW, VSE, XNE, YT, KD, HC, and J. And followed by ZIS A and ZIS F recombinase genes. So here the most important gene KD, which forms the component 1 alpha and beta, and H forms the component second. And SUM for the activity of these component first, second, and component 2 genes. And A is the positive control gene. I mean, when this gene will recognize that there is nitrogen crisis and it will switch on all other genes. And L is a negative control genes, which only looks at the level of oxygen, intracellular level of superoxide. If level of oxygen is much, it won't permit switching on the like it's the powerhouse of the main power point. So if it is switched off, the main switch is switched off, then even if there is nitrogen crisis, NIF-A cannot switch on all the genes. And how does NIF-A works? It gets signal from the upper tier, that GS, GOGRT, GDH, ADH genes. Means if a lot of nitrogen is there in the form of ammonia in the atmosphere, then these 4 primary emitting enzymes will be busy in utilizing this ammonia and converting these into amino acids. And by that time, it will stop the NIF-A to work because this will be a reflection that we have enough of ammonia. Don't go for nitrogen fixation. And no sooner the entire ammonia of the environment is fixed, consume this, then this GS, GOGRT, GDH, and DH will be itself. NIF-A will be free and to switch on the entire machinery to start biosynthesizing nitrogen. Provided NIF-A is giving signal that yes, oxygen level is under control. So this is the whole molecular genetics. So the function of everything is there for the Q gene is responsible for hemoptake, BV, N, and E for synthesis and insertion of FEM cofactor, F and J are reductor supply, F for reduction through pyridoxine, J for reduction for flavodoxine, Z is a promoter side, other things I have seen, and W, X, Y, T, C have unknown function till now. This and this F I will deal with. This and this F. This A and this F are recombinase genes. Wherever the NIF gene components are not in continuous units. First of all, under nitrogen classes, this A and this F will be switched on. These form a loop type structure and remove the intervening sequence so that the NIF gene becomes a continuous unit and NIF coding starts, nitrogen is formed. So this A is for 11 kbp, up to 11 kbp intervening sequence. And if this F is responsible for removing up to 55 kilobase pair of intervening sequence. And no sooner enough nitrogen fixed, again this and this gene are back, intervention sequences are back. And so is in rhizobia, the NIF genes are flanked by on the plasmids by 10 node genes. Again, in other species, two clusters, NIF gene is flanked by fixed gene means GLN genes or NTR genes in one or two second clusters. You see, now other important thing is that nitronase enzyme is not only able to break the triple bond between two N atoms, but also triple bond wherever present between two C atoms or between one C and one N atom. That's why as it reduces nitrogen into ammonia, it also reduces azide. Azide is very toxic.
Sir,
It causes
Botany
several
with
lysis.
LNMU,
But
it's
nitrogen fixing cyanobacteria. Can you switch off your mic? There's a noise. Can you please switch off your mic? There's a great noise. So azide also has triple bond between two N atoms at one place. So this is also accepted by nitrogen, broken into ammonia, and that's why there's no more toxicity of azide. Based on this, we have created azide-resistant mutant, which is able to fix nitrogen even in the presence of nitrogen fertilizers. Even potassium cyanide can be detoxified by the use of nitronase. And similarly, two triple and bond between two C atoms, acetylene has been reduced to ethylene, and this is the discovery based on which gas chromatograph instrument was
सर
devised.
आवाज गूंज रही है.
So someone has unmuted the mic. Please, I'll ask everyone to mute their mics.
Sir, I am just contacting them. Sir, वहाँ
Please
वाला
mute the mic. The administrator should mute the mic of all,
उसको ऑफ
except
करवा
me.
दो, उसका इको है प्रेजेंटेशन में सुनाई दे रहा है सर. यस सर, बहुत ज्यादा. वो बॉटनी पीजी एलएनएमयू का माइक को, हाँ सर, अब हो गया, म्यूट हो गया सर.
हाँ, तो ये पहले कर देखना चाहिए था आपको ना. Anyway, so we produce the de-repressed mutant of sodium azide resistant. What happens that in the nitrogen supplemented medium, nitrogen cannot be activated, but we have to make azide resistant. So in nitrogen supplemented medium, azide was also to put, and Nostrum muscorum, a cyanobacteria, was there. So in order to save one's life, a mutation was there that's a kind of spontaneous mutation. So that NIF gene was activated even in the nitrogen supplemented medium. And what happened that nitrogen came and it detoxified the azide. That's why we got one cell out of 10 to the power minus 9 cells introduced. And that one cell was later mass cultured and supplied to farmers to be used as de-repressed mutant of cyanobacteria, which fixes nitrogen even in the presence of fertilizer nitrogen. Otherwise, no sooner you add urea or ammonia, the inherent capacity of nitrogen fixation is stopped as an inherent quality. So the target of genetic engineering. Now we have the target to transfer to the eukaryotes, to the plants. But the time is very short. I don't know how to cover, but I'm trying to do it. NIF gene was established in 1975 in Nostrum muscorum while working in University of Dundee, Scotland. That NIF gene can be transferred unit. The wild type strain was heterocystus and nitrogen fixing. Its mutants were first isolated, which were streptomycin resistant and NIF minus, and both were mixed. And out of mixture, a conjugation happened to produce streptomycin resistant heterocystus and nitrogen fixing mutant. Either there must have been the spontaneous mutation for streptomycin resistance or a spontaneous reverse mutation for nitrogen fixation. But the frequency was noted separately to be very low, 1 to 10 power minus 8 to minus 9, whereas the recombinant was formed with the frequency of 1 to 10 power minus 4, means 1 out of 10,000. So certainly it was due to a recombination. Since NIF gene is only 24 kilobase pair long, whereas streptomycin resistant gene is 107 kilobase pair long, so the chance is that a NIF gene has been transferred. This was the first proof in 1975 the NIF gene can be transferred. The whole episome was worked out to be nearly same to that of Escherichia coli conjugation through the formation of tube. All genes have been mapped, and these are there. 25 genes are there. Of 0.25 million molecular weight, or as compared to the 10 million molecular weight of the main cyanobacterial chromosome. Based on same principle, the intergenetic transfer was also introduced from Anabina doliolum to Plectina boreonum. And then we also got transgenics in Escherichia coli, that is through M13 vector technique, shuttle vector, between Klebsiella and Escherichia coli. We could excise the NIF gene from Klebsiella and transfer it to Escherichia coli. This strain is preserved now at American Type Culture Collection Center. Anyone can have it for experiments in the University of Maryland. Now, out of many components of NIF genes, NIF-H in red color is the one gene which is almost in every neutron fixer invariably present on the same position with the same size, and it's never absent. I mean, other genes, one or two may be absent, but NIF-H is universally present. This is a very important discovery. Now, the first evidence of transfer of NIF gene to eukaryote was obtained by Salazar group and Postl group in yeast system, because yeast has a working plasmid. And at metaphase, it unites with the chromosomes and exchanges some of its DNA with the main chromosome. So that's why people thought that why shouldn't we introduce the NIF gene in the yeast working plasmid so that it may deliver to the main chromosome. But there was no insertion site in the yeast working plasmid for this NIF gene. So it was first introduced in Escherichia coli, and the yeast working plasmid was befooled in a sense that it's thought that it is receiving the part of DNA from Escherichia coli, and in between the part, NIF gene was already inserted. And that's why this NIF gene was very successfully engineered in the chromosome of yeast system. But this could not work because yeast is a very small system, just like a bacterium, although it is eukaryote, but it cannot jeopardize its work by transferring its lot of molybdenum and iron for nitrogen fixation. That's why it was transported in Nicotiana leaf in the chloroplast based on the Margulies theory of endosymbiotic origin of chloroplast, and the system that Anabina azole, a prokaryote, is present as well as dorsal leaf cavity. Both chloroplast and the cyanobacterial cell have similar kind of a structure, means free thylakoid and free DNA. Based on this, microprojectile bombardment of NIF genes from Klebsiella was made, and it was successfully engineered in Nicotiana leaf chloroplast. But the problem here was that chloroplast is the site of oxygen evolution and site of photosynthesis, so it could not work here. See the homology between cyanobacterial genome and chloroplast genome and how the genes were engineered. Complete set of NIF genes were engineered, but they are not functioning. So these are the various cultures we have in our laboratory of various kinds of Anabina, Gliotrichia, Solitoria, Spirulina, Strigonema, Cytonema, Calothrix, Nostoc, Nostocomion, Olosara, Vestrelopsis, Tolpothrix, Cytonema, Nostoc, Anabina fertilissima, Anabina constans, Ephinothes, Sanothes, Nostoc. So these are, many of these are non-natural fixings, supposed to be, but we discovered these are all natural fixing. And this unique cyanobacterium, this is a Lingbia, was found on the bark of mango tree in BHU campus, which is UVB resistant, which is fatal due to ozone layer depletion if it comes, because its range is 220 to 320 nm, and the DNA absorption spectra is 254 nm. So it may directly damage the plants, animals, and human beings. But this plant, which we found this cyanobacteria Lingbia, has a pigment cytonemin under the control of particular gene. We have marked this gene and isolated the gene. This is a very good source material for transferring to other plants and animals for protecting it from ozone layer depletion. So this is the future line of action. Right now, I've covered many of the slides. The first line is that cloning and sequencing of other nitrogenase structural genes for evaluating in-depth NIF diversity and to develop strains with inactive NIFL or active nitrogenase with excess of oxygen. This is one line of action. The entire world is busy doing with this. That means some specific laboratories have been marked, including ours, inducing development of constitutively produced superoxide dismutase and catalase enzymes in chloroplasts, and sophisticating the future technology for engineering NIF genes in non-photosynthetic cells of organelles, like mitochondria, through introducing suitable insertion sites for an up-to-the-mark acceptability of NIF gene components. So I think I should end here because of the paucity of time, and I'd be happy to invite questions, if any. Hello. Am I audible?
Yes, sir. Yes, sir.
Yes, sir.
Yes, if you have any question, although I could have gone even for 1 hour more in utmost detail, but
Yes, sir, of course.
there was paucity of time. Sorry, I had to be a little faster. So if there's no question, you may send your question on my email later, because your other programs should not be compressed as mine was compressed. Okay.
सर निफ्ट क्या है?
Hello. Yes. Any question?
सर निफ्ट क्या है?
NIF gene is a gene which biosynthesizes nitrogenase enzyme, which I told in uttermost details in the very beginning also. That nitrogenase enzyme has the capacity to absorb the nitrogen gas from the air and to break its very, very strong triple bond existing between two N atoms, so that every free N atom can be joined with the hydrogen atoms, protons, to form ammonia fertilizer. Because plants are not able to have this nitrogenase enzyme or NIF gene, so they are dependent on the ammonia or urea fertilizer supplied, or ammonium phosphates fertilizer supplied synthetically from the external sources. So it demands a lot of cost, a lot of fossil fuel to produce, manufacture in the factories at the cost of 450 degrees Celsius of higher temperature using fossil fuel. So this is the importance of NIF gene.
Okay, thank you, sir. Thank you.
Hello. Hello.
Yes.
Out of so many blue-green alga, which alga has the highest rate of nitrogen fixation? Like nostoc, anabina, boswellia, and so many alga. You have done.
Holosara is the organism which has a very high rate of nitrogen fixation. But then its culture in Holosara, but its culture in mass, Holosara, AULOSIRA, yeah. But its culture is not so fast as that of nostoc and anabina.
But in case of boswellia prolifica, the nitrogen content is highest among all the blue-green alga.
Right. Fixation rate. Fixation rate is assessed through gas chromatograph, which I said, based on the principle of the nitrogen's capacity to reduce acetylene into ethylene. like it reduces nitrogen into ammonia. So based on that device, a serine reduction technique, the gas chromatograph was devised. So there you put any organism and it will say which one is more efficient. Means not only the amount of nitrogen, in what time. Suppose in 1 minute it produces a lot of nitrogen and the other one will produce the same nitrogen in half minute. So the second one will be treated to be more efficient. So that is the parameter of how to assess the efficiency of an organism. Its fastness, yeah.
So in case of non-symbiotic nitrogen fixation,
Yeah,
non-symbiotic nitrogen fixer bacteria or blue algae,
yeah.
what adaptation they took to do the concentration of oxygen low?
Yeah, in symbiotic forms, the like rhizobium, the natural fixation rate is very high inside the root nodule. It is protected a better way for oxygen.
How they lower the content of the oxygen because nitrogenous enzyme is
Yeah, I have already explained everything. Like in a vector system, in cyanobacteria, heterocyst is there. Then in non-nitrogen fixed form, a gene, TS gene is there, the known as temporal separation gene. So whenever there is nitrogen crisis, the TS gene manifests, is triggered, and it stops photosynthesis for a while. So there is no oxygen evolution. And meanwhile, NIF gene is biosynthesizing the nitrogen. And whenever there is enough of nitrogen, then TS gene gets back.
okay. Thank you, sir.
I have illustrated these
Excuse me,
things.
sir.
Yeah, please.
Sir, I have a question.
Yeah.
Sir, why only heterocyst can fix a nitrogen in environment? Why vegetative cell in the filament cannot be fixed the nitrogen? Why?
Yeah, vegetative cells can also fix. So, but provided this has TS gene, like Oscillatoria. Oscillatoria has no heterocysts. They are non-heterocious forms. Non-heterocious forms have only vegetative cells and they have NIF genes. But then there should be a TS gene, means temporal separation gene. If that gene is present, like in Oscillatoria and Plectronema, it has been established. These genes stop photosynthesis for a while, temporal separation gene. And in that case, NIF gene is triggered. And even in the non-heterosexual forms, nitrogen can be fixed. But those organisms, those heterocysts are there, those genes do not work because heterocysts are already there, which is complete hyaline structure. What happens is whenever this A is active, it cuts off that loop formation, the photosynthetic photosystem second gene, where oxygen is evolved. So it is non-photosynthetic, but still photosystem 1 is present, so ATP production is there, and since there is no at all oxygen, so a lot of nitrogen is produced and nitrogen is fixed. So there is no need of temporal separation gene in the vegetative cells. Otherwise, in those levels where only vegetative cells are present, no heterosis, TS gene is manifested. So this is the difference.
Some more questions from
Sir, what are the problem in a aerobic and anaerobic condition to heterocyst fix a nitrogen?
What happens that in aerobic condition, the oxygen dissolves with the aqueous medium, means the water present in the cell. 90% is water. And then it converts to superoxide radicals. And that superoxide radicals completely blocks the nitrogenase enzyme already produced. I mean, irreversibly destroys it and stops the NIF gene because NIF-L is a negative control. It works on NIF-L so that it switches off the entire machinery, all 23 genes. So this is the problem with the aerobic forms. But in anaerobic forms, there's nothing like oxygen, means the threshold level of superoxide radicals is below the damaging level. So that's why NIF gene is always protected there. Nitrogenase is evolved and it works well.
Thank you, sir.
Sampan. It was a very nice presentation. And if anyone has a question, they can send it to my email to Dr. Sampan and he will answer. Because
Yeah,
we are running short of time. What?
Anytime you can use my email for putting questions.
Okay, thank you.
Okay.
So we are
Thank you very much.
short of time, and the new technical session is going to be started, and the chairperson is Dr. S.N. Chaudhary, Professor S.N. Chaudhary of this department, and the next speaker is Naveen Manakarmi from Nepal. Okay, sir. Thank you.
Thank you.
Just a minute, just a minute.
Okay, can I present?
No, just a minute. He is calling you. The chairperson is calling you. Dr. Naveen Narayan Manakarmi, welcome in the University Department of Botany seminar regarding the Naveen Narayan Manakarmi completed his master degree from Trivandrum University, Kathmandu, Nepal. He had 8 years job experience in the University of Nepal. Manakarmi attended several national and international conferences. He has published 8 research papers, and the research work are going on. The topic of the talk: evaluation of air pollution tolerance index in plants growing along with the Indian ring road of the Kathmandu, Nepal. Thank you. Now,
Time limit.
within time limit. You are further requested to complete your talk within the 30 minutes. That is the time allotted you. Okay? Go on.
Yes,
Okay, sir. Thank you. Thank you.
Okay? Go ahead.
Thank you. Namaste from Nepal. Enter screen. Is it visible? Does my slide visible?
Yes, yes, yes. Yes, it is visible.
Okay, is it visible? Okay, thank you. My
बहुत हो रही है
okay, my presentation title includes the evaluation of air pollution tolerance index (APTI) of plants growing alongside inner ring road of Kathmandu, the city of Nepal. Here includes my contents, my team member, research team members, introductions, methods, results and discussions, and followed by the conclusions. These are my research team members, Sabina, Sudarsana, Sakib, Susan, Janu, and myself. And talking about the introductions, like various air pollutants like heavy metals, particulates, polysaccharides, aromatic hydrocarbons, toxic gases, which only not affect the poor air quality but also human health activities like respiratory and cardiovascular disease. And around 90% of people in the world live in the poor air quality as determined by WHO guidelines. And such quality of air is degraded from the emission of motor vehicles and industries. And these are the serious problems for developing countries like Nepal. And in addition, our research focus not the whole country, the capital city, that is Kathmandu, which has bowl-shaped topography and very dense population and increased use of automobiles. So this makes the Kathmandu city most polluted city in terms of air pollution. And talking about vehicles, the number has been very exponentially increased. This is just an data from 2001 to the 15. That is like around 15 years data. Vehicle registered from 24,000 to more than millions, 7 millions. According to Environmental Protection Index, air quality of Kathmandu was ranked at 145 position out of 180 countries. And it is very difficult to reduce the number of running vehicles and to bring them up to date with the high maintenance standard, as due to the developing countries and lots of scenarios, it is very difficult to reduce the vehicles. But a pollution can be reduced or minimized to certain level by vegetation, as we are biological scientists. So this pollution can be minimized or reduced by the use of vegetation along the roadside of polluted area, and this is considered as a best.
Mr. Naveen, your slides are not sharing. Please share your slides.
Slide is not sharing.
Yes.
Now it's okay.
Okay. Uh, the pollution, air pollutions, that can be reduced or minimized in terms of biological activities or bio-based activities, that is plantation. And this method is called uh, passive control tools. And for this, we have assessed APTI, which stand for Air Pollution Tolerance Index. And for that, we have to calculate different components like leaf pH, relative water content, chlorophyll content, and ascorbic acid content. In our studies, we have randomly selected 13 plant species in polluted and a controlled site. And this study may help in selection of air pollution tolerant plant species and may contribute to maintaining the green belt inside the Kathmandu Valley. And this is our materials and methods. This represents the Kathmandu Valley. It is just like in a big pictures. This is GIS-based map, and this includes the Kathmandu Valley. And this is our study site. Actually, we have compared two different things. Like one is in the control site, which is less polluted. It is in the Godavari, where the botanical garden is situated, control site, and next in the experimental site, where the high traffic mobility is visible. That is range from the Tinkune to the Gausala. This is Nepal, and this is the slides. And these are the plan which we have chosen for the experimental purpose. And for the analysis of APTI, these are the 5 different components. And APTI is calculated by this formula. Okay. And now this is our results. And our result shows the ascorbic acid content, chlorophyll, pH, relative water contents, and corresponding the PI, APTI. And P represents the polluted site, that is Tinkune to Gausala, and C represent the control site. As the pollution increases, ascorbic acid content increases due to its antioxidant activities, and our result correlates with this. And similarly, photosynthetic ability will be decreased due to high pollution rate, and this also the indicator from the polluted site. And pH will be increased. Likewise, relative water content will be increased. And we have also done regression analysis between APTI and the corresponding components. And this is the regression analysis between APTI and biochemical parameters. And this shows there is significant and logarithmic relation between the APTI and relative water content. That means increase with relative water content is directly proportional to the APTI. And likewise, such kind of smooth, but not like the relative water content, such kind of relation is shown between the APTI and ascorbic acid. And likewise, for the pH, there is also a significant relationship, but for the total chlorophyll content, this is a negative relationship we have found in our results. Okay. And APTI is an indicator to describe the ability of plant to survive against air pollution stress by controlling pollution-induced problems. Plants with higher APTI values are tolerant to the air pollution and are used as a sink for air pollution in the populated site. So those plants which have higher APTI value can be used as a sink, as a biological or passive control for controlling the air pollutants. And plants with low APTI are sensitive. That means we can be used as an indicator. In our experiments, we found all of our plants that is 13 plants selected has APTI below the 11, which is sensitive indication. So this plant indicates use as indicators. And talking about the conclusion, we have described a very simple and low-cost technique to biomonitor urban air pollution. By means of the sensitive plants, by means of the moderate sensitive plants, we have described these things. And different parameters of APTI behave differently with the plant species in the present studies. As we see, APTI is directly related in case of total water content, but indirectly related with the case of another quantities. Ascorbic acid and relative water content provided greater tolerance than other parameters. So while calculating the APTI, the major role is from ascorbic acid and relative water content in compared to the pH. All the plants were sensitive to the air pollutants and thus are effective indicator of air pollution along the major road of the Kathmandu Valley. So our study shows that a selected area in the Kathmandu Ring Road from Tinkune to Gausala is polluted by means of the indication from sensitive plants, and development of green blend along the roadside with the plants having high APTV value is advisable. So what we recommend is like those plants which have high APT value. This includes the plants like Populus deltois and like Mosandrus grandifolia, Eucalyptus alba, and Ficus benjamina. These are the plants which have the high APT value and is feasible in terms of climatic condition for Kathmandu Valley. So we suggest these plants for treatment or for reducing the pollutions. Planting these species can reduce the air pollutants by different biological mechanism and can be cost effective for Nepalese government compared to the catalytic converters. Actually, the other physical methods for reducing the pollution is fitting the catalytic converters to the vehicles, which has obviously huge cost and huge investment, and probably the government might or might not be success to make catalytic converters fitting into the existing vehicles. But by means of biological scientists, we have provided the very alternatives and a natural way of planting APT, high APT value plants like this for these things. And thank you. This is my very short presentation, as our time has been very gone. So I've given presentation very shortly. Thank you. Thank you. Any question? Thank you. As time has been running, so I explain my research in very short time.
Sir, Naveen sir has completed his presentation. So if there is any question there, then they can ask or we can invite other speakers.
Is there any question?
One question. So Naveen Narayan, one company, he has taken all together 16 plants. Whether all they are the annual or the perennial. And the second question is what was the age of the plant?
Hello.
Mr. Naveen, the question is, which type of plant you have taken?
Any question, please?
Yes, which type of plant you have taken? Annual or perennial? And what was the age of the plant? Are you listening me, Naveen?
Any question?
Yes, sir. Dr. Naveen, you are listening me. Huh?
Hello.
Yes, sir. Am I audible, sir? Nabin sir, am I audible? I think there is a network issue there. He is not capable of listening to us. What are the questions there?
Okay, anyway. Thank you. Thank you. The next speaker. Okay, आप बोलिए.
Dr. David Mati Naika from Sri Lanka.
Dr. Jan Pathi, Naika.
From Sri Lanka.
I haven't heard any sound. Hello?
Hello, can you hear me now?
Yes.
Hello?
Yes,
Right, okay, thank you very much.
Naika.
Thank you very much. It's a pleasure for me to
It's
be in this particular,
Dr.
and I thank
Chairman of the session. I welcome you on the behalf of the Botany Department, LNMU University, Darbhanga. Dr. Jayan Pathi
yes.
completed his MBBS, MPhil in Cancer Genetics, and PG Diploma in Biotechnology. Currently, she is working as a senior lecturer in Department of Anatomy, former head of the department, Faculty of Medicine, University of Darbhanga, Sri Lanka. Her talk, The Understanding Molecular Locks and Keys, a way forward in nanomedicine. Thank you. Now you start.
Thank you very much. I thank all the organizers for inviting me, and also my special thanks to Dr. Gopalji, who has given me this contact and got me the opportunity. Now I'll start my presentation. I'll share my presentation. Can you all see my presentation?
Yes.
Right. Okay. So my particular topic today is molecular locks and keys, a way forward in nanomedicine. Why am I talking about this? Okay. Why am I talking about this particular topic? Okay. Let us see when we look into the history. When we were when penicillin, Alexander Fleming discovered penicillin, we were actually more than happy to see what is happening there in the medical world, because it has provided a better solution for the infections. And similarly, Edward Jenner, when he discovered vaccines, these are milestones in medicine. And then came a lot of other inventions, like the first discovery of X-ray, radium, polonium, discovery and cancer treatment, chemotherapy, and various discoveries were there. So we thought that the problem was ending actually, but we have encountered many problems thereafter. Antibiotics. So the discovery of antibiotics, we thought it is the end solution to the infections, but we were encountering various problems like resistance to therapy. And there may be, when it comes to various forms of drugs, the drug discovery is a lot of side effects and adverse effects. And talking about vaccines and treatment methods, there are variations among people. You can see the response of the patients will vary. And sometimes in imaging methods, when we are using, sometimes we may not be able to identify the particular lesion, especially at an early stage. And also, sometimes we might get undesirable immune reactions like anaphylactic shocks and various other forms of immune reactions, and failures in the treatment. For an example, like vaccine failure, what we are experiencing in even in with the recent COVID vaccines. So we were somewhere here actually when we are discovering the antibiotics and all. So then we were slowly climbing up in the ladder. Still, we have not reached the top. So we have to look into various other methods. So what were the problems we were encountering? So the initial milestones that we have gone through, our targets were very wide. And also, we were not targeting the fundamental sites, but we are going around it, but not to the point. And also sometimes our modalities in treatment, in management, in diagnostics, we are being non-specific. And also, another problem we have seen is the poor knowledge in that particular area of study. So this has actually restricted the future users of certain therapies or diagnostic techniques, or the limited users were there. So when we are looking at it, those are not sustainable actually. For sustainable development, we need certain things that can continue without problems or with minimal problems. And also, our main objective is to, in medicine, to save every life, not to leave behind at least one single human life. So each and every life matters to us. So when we are talking about the way out, actually we have to understand the basics, the fundamentals. So we have to get to the bottom of this. Because otherwise, the knowledge that we see at the surface is not sufficient to get into better precise targets. So we have to understand how the wheels are rotating. So we have to look into the big picture. So talking about life, the fundamental unit of life is at the cellular level. Although we were talking and we are looking at it in different levels of organization at multicellular level and in higher levels, but to get into the proper level, we have to get into the fundamental living unit. So when we are talking about the fundamental living unit, it is the cell, and it is a special macromolecular assembly. This is what we have to think of when we are talking about a cell. So those are special macromolecules assembled in a specific manner, especially in association with water. And this assembly is complex. Just to show you the complexity, I have shown here the molecular organization, the macromolecular organization of Escherichia coli cell. So this E. coli shows you, like E. coli is a simple prokaryotic organism. Now, when it comes to multicellular organisms, the eukaryotic cells, they are more complex. So, and these macromolecules are assembled in specific physical and chemical conditions in association with water, and they are very highly organized. And we have to understand that there is an inner aqueous solution that is being separated by the outer environment by a membrane. And there's constant interaction between the inner and the outer environment. So a cell is a dynamic unit. So, but although there are interactions, the inner environment kept stabilized. So homeostasis mechanisms are working in there. So there is these homeostasis mechanisms actually protect the status of life. So without the understanding of all these things, it is very difficult to get into precise targets when we are talking about disease and cures. So talking about understanding the cell and the cell, the fundamental unit, the living unit, you can utilize various modes and ways, but with the improvement of the technology, actually, you can see more details and more details. So it is important not to be at the superficial level. So with the invention of microscope, we know the microscope can actually the limit up to which that you can visualize things at a very micro level. But but when you look at the molecules, the cell is a molecular, macromolecular assembly. They are at a nano level. So you have to go beyond that, beyond the level of the microscope. So we need special equipment. So we were struggling with technology for a long time, but yet the amount of the wealth of knowledge that we were accumulating is vast, but still our targets were wide and non-specific sometimes. So it is important to get into the bottom level to understand the processes of life to get into specific targets. So here we can understand the subcellular level, the basis of diseases. So this is very important if we are talking about saving every life. So understanding the subcellular level, the problem at the bottom level is dealing with the sizes and properties of the things that we are dealing with, the targets we are dealing with. So they are at the nanoscale. So manipulating at this level now, visualizing them at this level is also challenging, and manipulating is more challenging. But implementing them at the multicellular level is extra challenging, I would say. So talking about nano level intervention, at a nanoscale, the properties of matter, if you look into the physical properties, the chemical properties, unlike when they are in the macro scale, at the nano level, the matter, the properties are changing. So we need to understand that. So sometimes the water in nanoscale tubules, like they were not freezing. You can keep it in the liquid level even at sub-zero levels. So the in vitro and in vivo differences we have to understand. Sometimes although we see a lot of things in vitro, we cannot implement them in vitro. So we need in vivo systems also when we are talking about it. So manipulating at nano levels in these levels are very difficult. So it is, but if we can understand these things and manipulate at these levels, it will expand the horizons to a greater limits. So we will have better modalities of understanding the basis of disease and treating them. I have included this slide just to show you the new technological advances where we can actually look into the nano level. So this light microscope converting into the nanoscale was a Nobel Prize winning invention actually. So it went beyond the normal limits. So you can use easily a type of a light microscope with a little bit of modification to visualize at nano level, and also the scanning tunneling microscope, which is used in chemistry, physics, and all. Usually, you can visualize the surface, and it will allow the manipulation of nanoparticles and atoms and small molecules at this particular level. So these technological advances will actually help us to get into the nano level or the smaller macromolecular level. So what are molecular locks and keys? If you look at the cell as the fundamental unit, you can see it has a limiting membrane. So what is a membrane? In general, a biological membrane separates environments, and it provides gateways because the entryways into the cell and out of the cell. And also when you look at eukaryotic cells, you can see there are little compartments inside the cellular organelles. So separate internal environments, separate by membranes. So these membranes are very complex structures in cells. So, and they are the gateways into the cells, into the organelles, and out of the cells. So it is very important to understand these things. So we can actually do a lot of things if you can understand these details and manipulating at this particular level. So let us look at certain examples. Before that, we will just look into what a cell membrane looks like. So the size-wise, it is very small. It is about 10 nanometer width. So visualizing it and studying it is not easy. And also, the more challenging thing is its compositional complexity. It is more challenging actually. It is kind of, if you look at, especially when you look at a eukaryotic, now even at the prokaryotic level it is complex, but when it is evolving into eukaryotic levels, it is getting more complex. And in multicellular type of organizations, you can see the tissue specificity of these cellular membranes. So although at a glance we might see that all the cell membranes look alike, but there is a immense diversity between various regions in cell membranes from sorry, I saw a message. Am I clear or not clear? Sorry. Am I audible?
Yes, ma'am.
Okay, because I saw a message though that I thought I'm not audible. Sorry about it. Right. So talking about the cell membranes, the immense diversity. So it is worthwhile studying this because there is wealth of knowledge hidden in these cell membranes, and it is the entryway to a cell. So manipulating that now, especially when we are talking about the diseases, there are a lot of diseases are connected to these gateways. And also when we are talking about the pathogenic entry, so nowadays we are worried about actually the infectious organisms, especially when we are talking about coronaviruses and all, because sometime back we were able to bring down the infectious disease morbidity and mortality. And then the non-communicable diseases were in the rise. But now again, the infectious diseases, especially from the virus world, the attack is kind of very challenging. So it is important to understand these things in order to find solutions to these particular problems, because our lifestyles, everything has been kind of changed because of this particular recent infection, the corona infection. So talking about the cell membrane, so it is the gateway into and out of the cell. So it forms a stable barrier between two aqueous compartments. This is very important to understand. So it is forming a barrier between two aqueous compartments. Okay. And also, when you are looking at the cell membrane structure, all the biological students are very familiar with this fluid mosaic model of the cell membrane. So talking about the things that are main particular things that are there, it is basically a lipid bilayer with the lipid molecules, the phospholipid molecules having a hydrophobic component and a hydrophilic component. And when it comes to animal cells, you can see the associated cholesterol molecules. And in both animals and plant cells, the cell membrane basically composed of lipids and proteins. And the heterogeneous distribution of protein molecules made it a kind of a structure that is not unique to each and every cell. And the distribution of other things like the carbohydrate molecules, you can see certain molecules. I have just marked them just to show you the diversity. So from cell to cell diversity is there, from tissue to tissue diversity is there, organism to organism diversity is there. So there are a lot of targets in this cell membrane. So studying the cell membrane might give us a lot of targets for managing diseases, even at plant world, even in the animal world as well. So when we are looking at the cell membrane, although it looks like one complete membrane, it is organized into many domains. So it is a very, that is why I said it is a very complex structure. So the molecular composition differs, and these molecules are basically involved in membrane transport, and as well as the smaller molecules can go through the lipid bilayer. For an example, water molecules might go through the lipid bilayer, and certain smaller lipid soluble molecules can go through, but whereas there are various So these are some of the, I have given some of the examples just to show you that where there are various domains. So these domains are, and maybe at micro level, at nano level, so various minute level of domains are there, which we have yet not understand many of those things. So just to show you the diversity within the domains, I have just taken the transmembrane proteins to show you how diverse they are in relation to their how diverse they are in relation to cell membrane. So these transmembrane proteins is one group of, if you go back to, you can see the transmembrane domains are one group of domains that are seen in cell membranes. So within the transmembrane group, you can have an immense diversity. So it is very important to understand that for the precision of to have precise targets in order to intervene at this particular level. So we will look at certain early efforts people have made in order to overcome certain disease processes. This amantadine is a tricyclic amine, which is used as an antiviral agent against influenza A virus, which actually inhibits the transmembrane domain of the viral M2 protein. So which is in the viral envelope, which will prevent the uncoating of the virus. So once the virus has been entered into the cell, it has to get uncoated for it to release its nuclear material, the genetic material, in order to get it incorporated into the nucleus of the cell and to produce the virion particles. So people are looking at blocking this viral M2 protein in order to overcome this. But unfortunately, it is a good antiviral agent, but there are a lot of side effects because they have where the cells that are in the human body, especially in the nerve endings, the amantadine has promoted the dopamine also get attached to the dopamine receptors to release dopamine. So when the dopamine is being released excessively at these certain nerve endings where amantadine was acting, they have kind of caused a lot of side effects like nausea, psychotic episodes like mania, hallucinations, agitation, confusion, which are actually undesirable. These are not desirable side effects. So these were a little bit of discouraging, but at least there is some kind of action is there against, or you can say some kind of it acts as an antiviral agent. So in severe infections, irrespective of these side effects, you can use it, but the uses are limited because of its adverse effects. And against intervirus B. And so let us look at some of these molecular locks and keys. I have just taken certain examples from the pathogens that are infecting human body. So let us look at the therapies that they have used earlier and now where we are. And we will try to look into how these locks and keys are working and how we can use them in order to advance the therapies that we are using at the moment. So you all know about Vibrio cholera organism, and the cholera infection was once, it was very rampant in countries like Sri Lanka, like India. So many deaths have been occurred. So the epidemics were there. So the main treatment modality was the use of antibiotics. So usually those days they were using tetracycline, and nowadays they're using chloramphenicol and various other antibiotics in order to overcome this cholera infection. But unfortunately, this very rampant use of antibiotics have caused the emergence of resistance, antibiotic resistance. And and also antibiotics sometimes can cause allergies. And also when you are using the antibiotics, another side effect is it might disturb the commensals in in that particular area of the body. So, and it might cause a super infection, and sometimes it can cause, even if it is in the gut, it can cause antibiotic induced diarrhea. So various other side effects were there, but still without any other, in the absence of any other therapy, antibiotics were life-saving. But these were the problems, the resistance, emerging resistance, and various other problems, the side effects that are coming up. So we need to look into various other ways of treating them.
Hello, madam. Kindly conclude. We are running short of the time.
Okay. Right. Right. So the better approaches were to. Hello. So I'm supposed to stop at. Sorry. I have 30 minutes, right? Hello. So I would just talk about this particular example only. So the main approach is it is the enterotoxin is polarized attached to a cell membrane receptor. So it will unlock the chloride channels in order to cause the leakage of material, the water and chloride ions, so causing diarrhea. So I will, because of running short of time, I skip my other examples. So there are better targets like blocking the receptors and the enterotoxin subunits. So various ways of unlocking them. But also we have pitfalls, because certain when we the pathogen is using alternative pathways. Researchers have shown that we are using alternative pathways. So there are various pitfalls and ways to understand those things in detail. So there are other benefits as well. Understanding this can So coming to COVID infections, you can see we can use these things in different ways, like ACE inhibitors. Now the COVID infection, they are using the ACE2 receptor, so blocking them, or to use certain receptor mimicking agents like decoy receptors to block the entry of the viruses. So there are various other modalities, because we can see there are a lot of effects in immunization as well. So understanding those things might help us to overcome all these problems. So, so if we can get into the bottom level to understand these things, especially those complex structures, you can get many targets, the precise targets. So if you, but the problem is if you don't look into the big picture, the various other outcomes, the various alternative pathways and other pitfalls, as we have seen in other diseases, these specific targets can make going to give us certain problems. But it is very important to get to the nano level of the cell as a way out in all these problems. Thank you very much.
Thank you, ma'am. Thank you so much.
Thank you, ma'am.
Any questions, please?
Thank you, madam,
Thank you very much.
for your nice presentation. And
Thank
because due to in this conference the time was very short, we will expect a separate lecture on some other time, and we will call
Thank
you.
you very much.
Okay. So on
Okay.
behalf of the
Thank you
head of
very much.
department, I congratulate you and I thank you for cooperating us. Thank you. Thank you very much.
The next speaker is from Bangladesh, Dr. Jawada Kun Meher.
Can you hear me?
Madam,
Okay.
Jawada Nahar.
Yeah, I'm Jobadatun Nahar.
Okay. Thank you. Jawada Nahar. Jawada Nahar.
Jawada Nahar.
Okay.
Welcome, madam.
Thank you, madam.
Regarding the Madam Nahar, Dr. Jawahar Nahar is working as Assistant Professor, Department of Genetics, Plant Breeding, Bangladesh, University, harnessing Dr. Nahar area of interest is temperature influence on plant proteome. Okay, you start, madam.
Yes, can you see my screen, like my presentation?
Yes, yes. In there.
Okay. Can you see my screen?
No.
Okay.
Screen को दिख रहा है?
Mm.
You start presentation.
Okay.
Please share your slide.
Okay.
Yes, yes, yes.
Okay.
Okay. It's all right.
Thank you. Okay, it's my pleasure to introduce so many Indian people at a time, and it's my pleasure to attend in this international conference arranged by Department of Botany, um, Latif Narayan Mithila University, Darbhanga. It's nice to meet. Uh, I also would like to thank you, Abhay Kumar, to invite me. Okay. Mm, I would like to present a topic. It's related to my PhD work. Okay. The title of my topics is Change of Protein in Sweet Cherry Flower Bud Upon Temperature Change. And this research is um, funded by DFG, Humboldt University, Yusuf Jamil Scholarship, and also the whole work is mostly the analytical work is done in the Humboldt University. So at first, I would like to thank you all this institute who support my research work. Okay, I would like to start with the background of my research work. You know that the climates are becoming more and more cruel to us, the planet, for the human being. And the change of climate is already showing so many disasters in different countries. Uh, Recently, we saw the flood um, in China, in Europe, and even in the India. So this disaster is because of the global warming. Okay, uh, the global warming is becoming more and more fast. And it didn't start now, it started several years before when the human civilization started. Okay. And the global warming or the temperature first effect on the plant phenology. Let me show how the temperature, global temperature is affecting on the plant phenology. You can see in my um, picture, in this slide, there are there are some peoples who are in the right side. It is the winter season. These all peoples are, I hope they are Indian. They are wearing so many warm clothes, even though they are enjoying or they are taking some heat from the fire. That means this warm cloth is not enough for them to support or protect from this winter. Okay. But did you ever think about that so many tree in the place where there are heavy cold, mostly in the temperate region and the polar region, long time there is snow and the trees are standing outside in the snow without any warm cloth or without any heat support. So the question is how these trees are living in this cold winter without any protection or without any warm. Okay, I can give you the answer of this is called the mechanism of dormancy. Okay, the dormancy is a mechanism which is introduced in the perennial tree species, flower bud, and that's why our annual crop they complete their life cycle before the winter came. But the perennial tree species they can survive in the winter because they have the capacity of dormancy mechanism. So dormancy is a mechanism when how when the plants are became in sleepy condition for the whole winter, and when the spring started, then the plants again started to uh, restart their physiological activity or their blossom. Okay, and this dormancy period or dormancy mechanism or the dormancy process started in the summer season when the flowers or fruits are in the peak in condition, peak in ripening condition. And it started with paradormancy. Dormancy is divided into three parts: paradormancy, endodormancy, and ecodormancy. Paradormancy is also called summer dormancy. It started when the fruits are in the highest level of ripening condition. Due to the growth of other part of the plant, the flower buds cannot grow. That's why it is called relative dormancy. And the growth of these flower buds depends on the growth of the other part of the plant. That's why I call paradormancy. When the paradormancy ends, the flower bud goes to the endodormancy period. And this is very important stage for the flower bud because during this stage, the buds transit or transmit from vegetative stage to the reproductive stage. And once the flower bud goes to this endodormancy, they cannot come out until they acquire a certain amount of chilling temperature. It completely depends on the plant species, even the variety. Different plant require different amount of temperature. Okay, even the species. Once the plant um, complete its uh, what called complete its chilling requirement, they come out from this endodormancy period, and this is defined at the T1. Hmm? When the flower bud completes its endodormancy period, then the flower bud is sensitive to the environment temperature. Whenever the bud get its suitable temperature, it started to blossom. Until this bud do not get its suitable temperature, it will not blossom. And this period is called the ecodormancy. During this period, he need a certain amount of forcing temperature to start or force to come out from this dormant stage. And this period is completely depends on the winter period. If the winter period is longer, then the ecodormancy is longer and the bloom will be late. If the winter is warmer, the ecodormancy period will be shorter and the bloom will be arise. That's why the bloom period of the bloom time of the flower in this plant species depends on the ecodormancy period. The global temperature, when rising, the winter period becoming shorter and the bloom becoming earlier, it is very dangerous for the flower bud. Even I saw in my professor orchard, only one night, the spring temperature went below the -2 degrees centigrade temperature for 2 hours. And at the afternoon, there was a snowfall for half an or 15 minutes. All the flowers of the tree was destroyed due to the cold shock. And the yield loss of that orchard was completely 100%. So it is important for us, the ecodormancy period. When the temperature started to rise in the spring, the first the flower bud started to rise by rising the level of water temperature. Then this period is defined as the T1 star. So T1 to T1 star is very important period, the ecodormancy. However, whatever happens from this paradormancy to the T1 star or the swollen bud, we cannot see anything from the outside. That's why we cannot define which one is ecodormancy, which one is paradormancy. Okay? So global temperature is shifting the phenological period in that way. And the ecodormancy period is very important for the flower blossom. Different phenological model has been developed to determine which time is suitable for the um, T1 or T1 star, the period of ecodormancy and endodormancy. But this all the phenological models are developed according to the visual observation. Very rare physiological mechanisms are concluded, but it's important to include the physiological accuracy of this model. My professor, Frank Slashky and Gauss, 2017, they published a paper. They developed the model of the sweet cherry flower bud. And this model is developed according to the visual observation of several years data. Sorry. According to their model, the sweet cherry for summit needs 41 chilling portion for endodormancy release. And at T1, that means the endodormancy release about 30 November. And the beginning of ontogenetic development, that means end of the ecodormancy, and the flower bud started to regrowth, it happened near about 21st February. So the duration of endodormancy is 24 days and the ecodormancy is 83 days. But this duration varies about 20 days. It's strongly varied and depends on the winter period and the winter temperature. Hmm? Many researchers, they observed the T1 start, that means beginning of ontogenetic development, according to their visual observation. But it observed that before 2 to 5 weeks before this T1 star, the water content started to rise. That means only the T1 star is not the time when, that means visual observation is not enough. 2 to 5 weeks before the visual observation, physiological activity of beginning of ontogenetic development starts. So phenological model observation base is not enough. This model must be, must be physiologically validated. That means you have to explain by the physiologically so that what will happen in the future temperature, we can predict the model. Okay, so what are the challenge of these models? So correct estimation of the duration of ecodormancy is the challenging for phenological model. Okay, blossom times mostly depends on the period of ecodormancy. So we have to find out the period of ecodormancy if we want to know or predict the which time is uh, can be suitable for the flower time. And to robust the model, we must these two parameter T1 and T1 star should be physiologically accurate. So sweet cherry is one of the most important economic fruit. I think in this audience, in this conference, every people like this sweet cherry. I didn't find anybody who don't like the sweet cherry. It has highly nutritious value and also different types of antioxidant, anti-inflammatory compound. Okay, but the proteomic study of these sweet cherry orchards is very limited. That's why many information of the sweet cherry dormancy, especially in the physiologically, is not known what is happening during this T1 to T1 star, which time T1 completed and which time T1 star, how these two stages are working, what physiological activities are active during this stage is rarely understood. But it is important for understand the influence of temperature during this ecodormancy because the ecodormancy is mostly depends on the temperature of the environment. So why I make this study? The aim of my study to determine the duration of ecodormancy in sweet cherry flower bud by the proteomic study. To do this study, I have identified the proteins for determining the T1 and T1 star. I made a correlation with the protein and the air temperature, and we compare the T1 and T1 star between the protein abundance and the classical approach. Classical approach is the way that means in classical approach we find out the this T1 in T1 star by the visual observation. And in the protein, in proteomic study, we identify the protein and find out some protein which can determine T1 and T1 star. That means the period of ecodormancy. Then we compare the classical approach and the proteomic approach to see whether these proteins can explain this period of ecodormancy. Okay, we use the our material was sweet cherry flower bud and the cultivar was summit. Here in the picture I can show you the sweet cherry flower bud. It contained many in one sweet cherry flower bud. It is bud cluster. One bud cluster contains 5 to 6, even 7 bud, including a central leaf bud. We collected the sample in whole bud cluster and we removed this leaf bud. After collecting the sample, we preserved it in the liquid nitrogen. And the experiment was conducted in the sweet cherry orchard in Berlin, Belgium, where the annual temperature was 9.9 degrees centigrade temperature and the precipitation was 562. We collected the sample for 3 seasons, 2012 to 2014-15. And we collected the sample for 3 trees. And from each tree, we collected randomly 3 flower bud cluster. The sample collection was done from October to April, 7 days interval, until the first visual sign of the flower, when the flower bud started to swell. When the flower bud started to swell, we collect the sample according to their visual observation. Swollen bud, side-green, green type cluster, and open cluster. After collecting the flower buds, we extracted the protein by the method. This method was developed by the Frank York Fetke Laboratory in the Potsdam University. I have modified this protocol according to my sample, and we collected we extract the protein by using this protocol. It's a HIPPIS EDT extraction buffer and isolated by one-dimensional SGS page. After collecting the protein, we have identified the protein by merit of analysis. In this analysis, the proteins are digested by the trypsin enzyme, and we use the HCCA matrix to identify the protein. Mass fingerprinting was done by collecting the mass charge ratio, and the database Swissport and NCBY database was used to identify the proteins. After identification, we analyze the volume intensity of this protein by the ImageLab software. The volume intensity is the color intensity of the each protein band. We collected the data of each protein band, the number of band, the color of band, and the molecular weight of this band. Now, I told you that before that according to the protein, we determine the time of T1 star and compare with the classical method. In classical method, leaf fall, T1 star, swollen bar, this data was collected by the Smilashki and Gauss. Leaf fall is the time when the 100% leaves is fall down from the tree. And T1 is the time when the endodormancy ends. And this is determined by the climate chamber experiment. This is the picture of the climate chamber experiment. When we collect the sample, we also collect a twig from the tree, and we put it in a water containing bottle, and we put it in the normal room temperature, so that the plant, when the I inform you that when the endodormancy ends, the flower bud becomes sensitive to temperature. When he gets the suitable temperature, he started to blossom. But before the endodormancy ends, even though it gets the suitable temperature, it will not blossom. So when we put the uh, uh, twigs in the normal temperature, and the twigs which are completed their endodormancy period, they started to blossom. That is the indication of the T1. And T1 star, I inform you that T1 star we determine by the increase of water content. That means before the visual observation, 2 to 5 weeks before, the water content started to rise, and this period we determine as the T1 star. Swollen bud, side pin, grid tip, tight cluster, grid tip. These all are the visual change of the flower bud structure. We also collect the weather data from the bird in the island. We also collect the data of protein content and peptide intensity. After collecting the data, we analyze the protein band, significant change of this protein band by one-way ANOVA and Turkey SST test. We also compare we have make a correlation, Pearson correlation with between the protein band, volume intensity, and the air temperature. Okay, let's see what result we get from our research. We find out 66 protein bands, which are from the one-dimensional SDS page. Among the 66 protein bands, we could identify 33 proteins by MALDI-TOF analysis. The 33 proteins are grouped into different classical functional groups. Most of them are related to the metabolism of amino acid and protein, defense and stress response, phytohormone, secondary metabolite, and carbohydrate energy. And interestingly, we found that 4 proteins are changing significantly. One is neoxygenase synthesis, major chain protein 2AB1, rubisco, and phytosynthesis. These 4 types of protein bands, these are changes significantly during the T1 and T1 star. And these bands, we found these 4 types of protein. Let's see how these proteins changes during the period of paradormancy, endodormancy, ecodormancy, and ontogenetic development. Most important result we found in case of neoxanthine synthesis protein. In this picture, we mark 3 different periods according to the classical method: paradormancy, endodormancy, ecodormancy, and ontogenetic development. This is the this is the season 2012-13. This is the season 13-14 and 14-15. Each year, the neoxanthin synthesis protein shows a similar pattern of change uh, during the endodormancy, ecodormancy, and ontogenetic development. During the endodormancy period, the level of this neoxanthin protein band increases, and the winter period, that means during the ecodormancy period, the level of these protein bands was nearly the same level. And ontogenetic development, they started to reduce their intensity. When we analyzed by the significant test, one-way ANOVA on HSD, we found an interesting result in the case of the protein band 33, that means neoxanthin protein. In 2012-13, this protein band was significantly higher in T1 to T1 star compared to the uh, endodormancy and the ontogenetic development. And it clearly indicates that after the T1 star, all the protein in volume intensity was significantly lower. In case of major allergen crew avian, this is the protein which have the defense responsive protein. It also showed the similar result that after T1 star, this protein band showed significantly different compared to the other period, leaf fall, that means paradormancy, anadormancy, and ecodormancy. We also saw that there is a Ruby scope, the volume intensity of this protein bands increased uh, increased significantly after the from the T1 to T1 star. Okay, now we can see what was the situation of the mean air temperature of this orchard during this 3 period. S1 indicates the time, that means the beginning of October when we started the sample collection. And T1 is the time the endodormancy release. And T1 star is the time when the beginning of ontogenetic development start. So if you have a look at the T1 star in 2012-13, is quite longer period. And for this reason, the uh, ontogenetic development was quite late. Then we make a correlation with the temperature and the protein bands. We found greater changes during this period. Uh, the uh, the changes of the temperature during this whole period was follow the changes of the protein band neoxanthin synthesis. With when the temperature was plateau, the during the ecodormancy period, the level of the volume intensity of neoxanthin synthesis was also almost the plateau. With increasing the temperature, the volume intensity of this neoxanthin synthesis also became reduced. We made the correlation and found that we make two different types of correlation. One is interseason correlation and another is within phenophase correlation. Within phenophase correlation, the temperature has strong correlation uh, from the T1 start to ontogenetic development. That means when the protein, uh, when the dormancy, ecodormancy ends, then the temperature becomes very sensitive to the flower bud. But the flower bud became very sensitive to the temperature, and it shows all the protein band shows significant correlation with the temperature. In case of interseason correlation, the neoxanthin synthesis protein shows very strong and significant correlation during the green tip. Also, we found the strong correlation with the during the side green, tricluster, and the T1. That means neoxanthin synthesis is strongly influenced by the temperature, and it has a negative influence. So at the end, we compare the classical approach and the proteomic approach. We compare and found whether which protein can be close to the classical approach, which can be explain the period of ecodormancy. In classical approach, T1 is in 2013, 12, 13, it was 332 days, 13, 14, 323 days, and mean was 332 days. When we compared with the neoxanthin synthesis, it was 14 days earlier, 14 days late compared to the classical approach. Other Rubisco and major cherry allergen group AB1, they cannot determine the time of T1. And T1 star, when we compare with the classical approach, it was only 2 days difference with the classical approach. That means the neoxanthin synthesis can be a good indicator for the period of ecodormancy. This is the summary of my summary. This is the result summary that we make a conclusion that neoxanthin synthesis protein can be a good indicator to determine the T1 star. Okay, this is the first step of the proteomic research in the Swiss cherry. Uh, very few research work has been done in the sweet cherry. Still, we have to do so many research to understand actually what is happening during the ecodormancy period. Actually, which time is the time, which time is perfect to to determine the T1 star, and what is actually happening during the T1 star. Mm. We have to find out the best marker to determine the T1 star, because many researchers has different view to determine this T1 star. Now we have huge research perspective, or we have to do a lot of research to find out the best way of to determine the T1 star. I would like to thank you, Professor Frank M. Slashki, to supervise me during the whole period of my research. Also, Dr. Klaus Peter Gauss, who supported me by his sample and also supervised me. I also would like to thank Professor Jörg Fetschke to support me in to work in his laboratory, and also I want to send my thank you to the University Latif Narayan Mithila University Darbhanga and the Department of Botany, and special thanks to the Abhay Kumar to give me a chance to make my presentation in this lecture. Thank you for your attention and welcome for any types of question. Please feel free if you have.
Thank you, madam. Your talk is very innovative and informative. Thank you, madam.
Yes, welcome. You are most welcome.
Talk is very informative and innovative. One question for you, sir, madam.
Sure. Most welcome.
You have taken the comparative study of the proteins. What about the hormone, dorming? What was the status of the hormone dorming?
I cannot clearly understand you. The sound is not clear to me. Could you please explain a bit more clear, loudly?
Loudly, good, sir. Ma'am, you have
yes.
comparative study of proteins in between T1 and T1 star.
Yes.
What of the hormone status of hormone dorming? Let me know. Have you taken or have you seen the status of hormone during the T1 period?
I cannot understand the question clearly.
Ma'am, he is asking. Hello.
Yes.
Are you listening me?
Not so clear.
Are you listening me?
Yes, I can listen you, but the sound is quite slow, not clear. It looks like you are talking so far away.
Your sound is clear. And he's asking that
yes.
You have studied about the protein.
Yes.
Protein. What about the hormone?
Hormones. Okay, hormones. Yeah, it's a good question. Yes, hormones. Yeah. Actually, my research group, they are trying to find out the best marker to determine the time of T1 and T1 star. They already studied about the nitrogen status, metabolite, hormones, uh, especially the abscisic acid, sugar level, ethylene, and other molecules. From this, they make a conclusion about their model. So my task was to find out if I can find any protein which can make the clear idea about the T1 to T1 star, so that combining all the result, we can explain actually what time is the perfect for T1 to T1 star, actually what is happening during the T1 star, which biological marker can exactly determine the T1 star. To make a clear picture, we we make also hormonal, especially the abscisic acid. Abscisic acid, we saw that during the time of the ontogenetic level, the abscisic acid is strongly reducing. They also do that. Thank you.
So thank you, madam. Thank you. I have taken all the parameters of this transition and metabolites. Thank you. We shall contact you later on for full lecture regarding your topic and next topic. Okay, thank you.
Thank you very much.
अब एक ब्रेक है तुम लोग के लिए आधे घंटे का
Thank you, madam.
नेक्स्ट
Yeah,
We are announcing break.
देयर इज अ ब्रेक now next session will start from 2 o'clock no 2 o'clock
Thank you, madam. Thank you. You have given the more time.
thank you. You're welcome.
ब्रेक हो गया आधे घंटे के बाद हम लोग मिल रहे हैं मोबाइल उठा लो
आवाज नहीं आ रही है।
हाँ जाएगा सर आवाज
The new technical session is starting after the lunch break. And in this session, we have the first lecture by Dr. Ramila Pathak, currently working as professor in the Department of Botany, Punjab University, Chandigarh. She is known for her selfless and dedicated commitment and significant contributions to scientific development and promotion of Indian orchids. She has been actively pursuing researches for the past over 2 years in diversity, developmental biology, micropropagation, eco-restoration ecology, and conservation of orchids. She has delivered invited lectures in several national and international conferences within and outside the country, including Japan, Malaysia, South Korea, France, USA, China, Nepal, Singapore, South Africa, Thailand, Twitter, etc. She has been conferred with TOSI Plaque of Honor in 2008 and Mrs. Usha Beach Memorial Award 2011 by the Orchid Society of India for her significant contributions to the growth and development of Indian orchidology. She is elected. She has been elected Fellow of Linnean Society of London in 2020, 2012 and member of IUCN, International Union for Conservation of Nature and Natural Resources, SSC, Species Survival Commission, Orchid Specialist Group during 2011. She is a member of the National Academy of Sciences. So we are really proud that a person with such high achievements has consented to deliver a lecture in our conference. So over to Dr. Pramila Pathak with a request that, that her deliberation should be of a duration of not more than 30 minutes. So over to Professor Pramila Pathak.
Thank you so much, Doctor, for your kind words. In fact, I must thank the organizers of this international conference to give me this opportunity to interact with the scientists from India as well as abroad. Thank you so much, Doctor. So, I'm going to take everyone today of the participants of this conference to the beautiful world of orchids. In fact, a fantastic world of orchids. And this, I would say, these orchids are the most beautiful flowers on this earth. And I have prepared this lecture basically taking in view of the young students and the young researchers to motivate them for taking the orchids as one of the research material for their researches. I always start with my lecture with my poem that in 6 days God created earth with man to rule it all. Of course, he made the mountains, forests, beautiful flowers and all that. But one day, one flower family caught his eye and caused a lot to say. Their blooms are surely regal and the beauty is supreme. For the different forms and colors constitute a poet's dream. Then God knelt down and blessed these plants, and tenderly he said, I'll call this family orchids, and its numbers shall spread to all the regions of this earth, and its bloom shall be the queen of all the flowers I have made, for none finer shall be seen. So orchids are Mother Nature's masterpiece. If we see the world, there are more than 600 genera and 30,000 species. And in India, there are nearly 155 genera and 1,256 species, according to a recent estimate published in a book published by the Botanical Survey of India. Now, before I start with the other aspects, I think I must explain what are orchids, how do they look like, and where do they live, what is their habitat, what is their habit. They are cosmopolitan, they are present all over the world. They are highly diverse, they are highly evolved, they are under active state of speciation. They are inherently slow growers, I must say. Like they have non-endospermic seeds. Like unlike other flowering plants, they have the seeds which are non-endospermic, very minute. They are produced in large numbers. And so with the result, that means the seeds cannot germinate in nature. If the seeds germinate, less than 1% seeds would germinate if they're fortunate enough to come in contact with a specific mycorrhizal association in nature. And if even these fortunate seeds which come in contact with the mycorrhiza and they germinate, so then from germination till the flowering stage, it takes 6 to 12 years for a seed to reach the flowering stage. So you can understand how inherently slow growers these plants are, and these are habitat specific. And post pollination development of ovules is again one of the characteristic feature for these orchids, as this is scientific reason, in fact, for the fact that they have long shelf life. You must have heard about that orchids have the flowers remain fresh for a longer time, starting from 1 week period to even 4 months. So this is a basic picture, I would say, scientific explanation, that there is a post-pollination development of ovules. I will not go to the detail, but I'll just speak. Just try to explain that what happens when in other plants, flowering plants, when a pollination occurs, then by that time ovule is completely developed, and embryo sac is there, and egg is ready. So the male gamete goes and the fertilization takes place and fuses with the egg and produces zygote. But here, when pollen lands on the stigma, by that time, that landing of the pollen on the stigma would give the stimulation in the ovary to for the initiation of development of the ovule. So what a great contrast, you can understand. So by the time fertilization occurs, it may take 3 months, 4 months, 5 months. So that is the reason the flowers remain fresh for a longer time. Then there are pollination related floral complexities. Again, this is a very important thing. The flowers look like bee-like, they look wasp-like, they look dancing lady orchid, toothbrush orchid, and they assume various shapes, and these are the complexities which I'm going to share in my presentation. Then there are ill-defined barriers of reproductive isolation. Again, this is a very, very important aspect. As we see, when any in the plant speciation occurs, then the morphological barriers of isolation and the reproductive barriers of isolation, they have to co-evolve. But here what happens is morphological barriers of isolations are there evolve, but at the same time, reproductive barriers, they do not co-evolve. That is the reason, So they look apart, and more than 1 lakh, hybrids, they are present worldwide, which are produced. The microtrophic, I told you that's a specific mycorrhizal association of these plants. Now, they may be epiphytic. In the epiphytic plant, there are structures like pseudobulbs, and their aerial roots, which are covered with outer tissue called filament, again a characteristic feature. But in the terrestrial plants, in the underground, you find tubers, and these tubers are very, very important, as these are highly medicinal. And what happens is these tubers, and these are dried and they're powdered, and this powdered form of these tubers is called salep, which is used for the preparation of various medicines, because besides their floricultural importance, they're medicinal important as well. Now, so they may be terrestrial, they may be lithophytic, they may be epiphytic, they may be subterranean. That means most of the life they live underground. I'll just show you these types of orchids as well. So in nature, they live in these are the orchids. They live in association with the certain other plants. Like here, they're living in a closely associated with the lichens. We really don't know as yet what kind of association do they have, what kind of exchanges are occurring between them so far. So this is a terrestrial orchid. This is called, common name is Salem Panja, and this orchid was collected from Kulu Valley. Um, it is occurs in marshy areas, and from Manali to downhill about 10 kilometers. And then its tubers, are very, very highly medicinal important, and they are sold at the rate of 20,000 per kg. So this is the plant which I wanted to show, that this in most of the life, it's a Rhizoanthella gardneri. This in most of life it remains underground, and only at the time of flowering the flower comes out on the earth. So this is an epiphytic plant. This I wanted to show how it is growing on the as epiphyte on the tree, and how the beautiful flowers it has. So this is Dendrobium falconeri. And then I wanted to show the diversity and the color shapes and the flowers. So this is yellow colored beautiful flowers of again epiphytic orchid, which grows on the trees. And here are the long cans, which absorb moisture from the atmosphere, and they conserve or store water for their survival. So this is Oberonia, again one of the orchid which occurs on the tree, and it lives in close association with the ferns. So coming to the flowers, as I told you, they're the most beautiful flowers on this earth. So you can see here in the picture. So first of all, I would say they have varied sizes, shapes, and colors. And labellum. So these are the characteristic features of these plants. The first feature is presence of labellum. Labellum is also called lip. So lip is, when in the colored petals, in the flowers, you all know there's a colored petal. So one of the petal gets modified to take various different shapes, striations, different colors and different shapes. So this petal is called labellum or the lip. And this is basically to attract different kinds of pollinators. And then the second feature is compound pollen. So they have a compound pollen, pollinia. Then there is a process of resupination. So resupination is very important phenomenon. Maybe the students of MSc may be interested to know, postgraduate students, that what is resupination. This is just a rotation of an ovary at an angle of 180 degree to just, if like there are let us say if there are 10 flowers in inflorescence. So the ovary gets rotated at an angle of 180 degrees, so that the beautiful lip portion of all the flowers, they come to lie towards the center, one place, so that they can attract more pollinators. Of course, there are some exceptions for the own non-resupinated flowers also, but then they are bisexual, and they are few unisexual as well. So I brought this slide to just show you that how the orchid flower looks different from the normal liliaceous flowers. This is normal liliaceous flower where you can see the normal 6 petals and with the androsia with the stamens and filaments and anthers. But here you can see this one of the petal gets modified to form the lip or labellum. And here you can see floral formula. I just wanted to show the students this zygomorphic, bisexual, 3+3 variant. Then there is a presence of gynostegium, that is male and female, like filaments and style. They are fused together to form a common structure, gynostegium, as informed in the Asclepiadaceae. And then androecium maybe 1, 2, 3, and ganoecium is tricarpillary syncarpus inferior ovary. So this is a clear-cut example of the resupination, which I just explained to you, the rotation of the nexus of ovary. And then this is just a fruit, how when the seeds come out, the fruit pus, how does it look like? Millions of seeds are produced in one capsule or in one fruit. So this is the pollinia. How do they look like? This is a fertile stigma here. And then the pollination. So you can see how these flowers take the similar shapes and the colors of the male insects of the flowers. So there is a phenomenon called mimicry. Mimicry is when the flower mimics various other insects or other objects. So this is a cleistogamy. And as far as pollination is concerned, that means many flowers remain closed throughout their life. Then there is a chasmogamy also, where they remain open, is the case in most of the orchids. Then there is autogamy in several terrestrial orchids, and most in the orchid there is a allogamy, that means cross pollination. Now, as far as attractants are considered, I must tell you one example, that is an Angraecum sesquipedale. The spar is this plant. It has a flower, has a spar, which is 27 to 43 centimeter long. And then at that time, Darwin said, after seeing that spar, that there must be some pollinator of this orchid who must be having a long, structure, to pollinate this orchid. So this later on, this proboscis, later on, this new pollinator was discovered, that was Xanthopan morganii in 1907, which had a long proboscis to pollinate this. So nectar is there, which is a form of nectar tubes and spurs, which they provide to the insects. Then callus masses are there with different colors, orchids, flowers, a bright red, orange, yellow. So depending upon these, they have different orders also. The sweet, pleasant, sweet. And then there are different pollinators like bees, butterflies, moths, birds, and flies. Now here you can see this labellum is like, you can see it's like a lady sleeper, so beautifully decorated. So this shoe is like a lady sleeper. This flower gives the shape of a lady sleeper. This is also called lady sleeper orchid. So you can see the color. This is called red venda. This is again one of the rarest rare orchid, already listed in the red list of plants or IUCN list. So this is red venda, which is called Renanthera imshutiana. So here you can appreciate, the color of the insects and the color of the flowers. How do they mimic each other? So here also you can see the male insects. What happens? This is a process again, phenomenon. I would say this is called pseudo copulation. Again, a very very characteristic feature of these plants. These plants are highly smart plants. Now what happens is, here the flowers of these plants start mimicking the female insects of the flowers. So when they mimic the female insects of the flowers, then the male insect comes and sits on these flowers, considering this flower as its female partner, sits on it for some time, and in a process it pollinates these flowers. So this flower looks like a flying bird orchid. You can see the flying bird with the long beak. So it has also a phenomenon where it tries to grab the insects, and then when the insect has to come out of this flower, it has to pollinate this flower. So it's again a very interesting orchid. It's called a bucket orchid. Now here the 3/4 of this bucket is always filled with some liquid or the smear, where when insect comes, just sits on the rim of this cavity, just try to smear its legs with this fluid and enjoy it. But in that time, it falls down in that bucket. And then if it has to come out of this flower, it has to pass through the passage where there are pollinia. So while coming out, it takes the pollinia along. You can see here, it's an insect. It's taking the pollinia along with it when it's coming out, so that when it goes to the other flower orchid, it pollinates. So how smart these plants are. They have developed their own mechanism to survive on this earth. So there are, of course, there are some seeds also. There's some pseudonectary, pseudopollen as well. Now here, you can see this is called dancing lady orchid. As you can see, arms, frog, arms. So this looks like a dancing lady orchid. Now this is like a cocoon. You can see here the hello. Hello.
Yeah.
Am I audible, please?
Yes,
Yeah.
you are audible.
Okay.
Yes, ma'am, you're audible.
Okay, so this is like a cocoon. So this helps, takes the shape of cocoon. So next is I had been to Miami, international conference. So here the one of the growers, he had brought this orchid, raised this hybrid, and I was really surprised to see that instead of one lady sleeper, it had two lady sleepers, on its flowers. So beautiful orchid, you can see. So here the process of pseudocopulation is highly depicted. You can see it's a flower which is mimicking the female partner of this male insect. You can see the male partner is sitting on it and the pseudocopulation process is going on. And here again, the pseudocopulation process, I wanted to show you how the female insects are mimicking the female insects of these male insects. So sometimes they're robbers as well. I mean, the spiders and all that. And then these flowers are also sometimes in the close association with the ants. I have seen this happening in my own greenhouse, that sometimes many ants are running here and there on the stem. So initially we were really surprised at what is going on. Then we really realized that these ants are helpful for dispersing seeds. pollen, and they provide nutrition also, and they ward off, help in warding off the herbivores. And in return, the orchids, they provide them shelter and extra floral nectar. I must say, all the visiting ants are not pollinators. So this is again a very interesting, orchid. This is called Schumbergia business, and it is called carnivorous orchid because we have studied many carnivorous plants in our middle class, 10th class, and all that. But it is very interesting that here it is stem. I have told you this is the bulbous stem. Here, when these stems get older, then ants start multiplying here, and they get shelter in these pseudobulbs. And just to prove that whether this plant eats, these ants or not, so a carbon labeled ants were given to this plant, and initially later on it was found that the nutrition was carbon labeled, so that was clearly indicating that it is a carnivorous plant. So this is, I wanted to show how its seeds look like. I told you the seeds are produced in millions in one capsule. If you open the seed, the seeds come out in the form of a powder, so they are highly microscopic and produce in large numbers. So maybe they're again adaptive feature to survive. So you can see here the seeds come in association with the mycorrhizal fungus, initial stages for the germination processes. And once the fungus enters the seeds, it starts moving to other different cells, and start other cells, roots also it enters, mycorrhizal fungus. And the association of the mycorrhizal fungus and the orchid plant is throughout, and it of course remains in the underground parts. Once it enters the root cells also, it forms pelotons. It's again a characteristic feature, of the orchids. So fungal endophytes may be Rhizoctonia, armillaria, and all that. So the mycorrhizal plants race may be healthier and higher net dry weight, rich in nitrogen and the mineral contents. Now, once these orchids were discovered, they were discovered basically for their medicinal properties, initially. So it was said, the tubers, I have already told you that terrestrial plants have tubers. So it was said that in case the shriveled tubers are fed to a expecting mother, she is going to deliver a female child. And if healthy tubers are fed to a expecting mother, she's going to deliver a male boy. So these are the facts and myths which were present in earlier times. But of course, these orchids are used for curing a large number of ailments, like they are used for curing rheumatism. So this is a orchid. These roots are used. Sometimes stem is used. So it is written in the bracket. They are used as a tonic. They are used to cure nervous disorders. They are used to cure digestive problems, to cure cardiac problems, as an expectorant, anti-cancers, and all that. And I must say that this Seraptalum maculosum is very interesting, and considered to enhance the uh, longevity of the person. So there is a Professor Joseph Arditi who has written more than 10 volumes on reviewed articles on the orchids. So he just tried to analyze the data of all the orchidologists throughout the world, and he reached a conclusion that the persons or the youngsters, whosoever, any person who is associated with the orchid researches and has an average life 10 to 12 times more than 10 to 12 times more than the other people. So I would say it's an open invitation for the young researchers to take orchid researches as one of the challenge. So
Please conclude.
Okay. So in Ayurvedic type of medicine, these are the Chyawanprash, it is used, this Rajivak, Rishpak, Riddhi, and Vriddhi is used. But all these orchids, are victims of their own beauty and utility. They are, from nature, they are being uprooted. There are large number of development programs and all that. So we are trying to work on their conservation programs. They look like smiling orchid. They look like pineapple orchid. They look like toothbrush orchids. So, I mean, they're this floral mimicry, which I'm like going to show to all of you. And not even they have left the Punjabi hippie to resemble, and a ghost, and orchid mania is there throughout the world. Everybody would like to possess the orchidaceous, any orchidaceous possession. So at the end, I would say there is a strong need to conserve these orchids on this earth, and there is a lot of work need to be done. And CITES also prohibits a trade of orchid species by placing certain species in Appendix 1 and Appendix 2. So we cannot export these, orchid species to other countries. We have to take special permission. And there is a tissue culture program by which we are trying to conserve them, mass propagate them on this nature. And all these things like orchids are considered as national flowers all also in many of the countries. And there's some promotional programs. We have Orchid Society of India. We are promoting their awareness, and we are publishing organizing national programs every year to create natural awareness amongst disorders. We are, some painting or competitions are organized among the youngsters to popularize it. And then we have published these two books also, and we are publishing these I would say international journal, the Journal of the Orchid Society of India and Orchid News. And as I always, when I conclude, I always say this orchid is a laughing orchid. We always say, though I have been associated with the orchid for the last more than 30 years, it is laughing at me for my ignorance about this orchid plant, because they're the highest, smartest plants on this earth. So we strive to understand, popularize, conserve, and propagate orchids. Thank you so much.
Thank you, Dr. Pathak, for dragging us into this beautiful world of orchids. I would like to put a question.
Yeah,
In
please.
our area, we are on the border of Nepal.
Yes, sir.
So in the bordering area of Nepal, there is a beautiful garden called the Garden of Raja Salhesh. And there is a beautiful orchid, Dendrobium ephyla.
Right.
That is called the love orchid, love flower. So history associated with this flower, and I think we would like you to throw some light on this orchid, Dendrobium ephyla.
Yeah, Dendrobium phylum is a highly, again, a medicinally important species, and it is of course growing in India as well. My students have done a lot of work on its endangered plant. I would say we have done work on its mass propagation and conservation also, and many of the plants we have conserved in the nature, and they're growing well and flowering every year. Yes, I thank you so much for.
We would like to have it in more details from you regarding this orchid. It is characteristic of this area of India and Nepal.
Yes, sir. Yes, sir. Yeah.
Acha, I would like the audience to put some questions, if any.
Yeah, sure, sir. Sure, please.
If not, thank you very much, and for joining us this afternoon. Thank you again.
Thank you so much.
Our next speaker is Dr. Ritesh Krishna. Is he here? Has he joined? Yes, welcome. Dr. Ritesh Krishna ji, very
Yes,
welcome.
I'm here.
Dr. Ritesh Krishna leads the computational genomics research at IBM Research UK. Dr. Krishna obtained his PhD in computer science from the University of Warwick and moved to the University of Liverpool for his postdoctoral research, where he was associated with the Institute of Integrative Biology, Center for Genomic Research, and the Institute of Infection and Global Health. Ritesh Krishna is an expert in interdisciplinary and is an interdisciplinary scientist working on the interface of life science and computer science. At IBM Research UK, Dr. Krishna is responsible for building scientific progress, collaborating and strategizing that are aligned with the needs of British industry and UK government's industrial strategy guidelines for life sciences sector. So over to Dr. Ritesh Krishna ji.
Hi, good morning, everybody. Just trying to share my screen.
Ritesh Krishna?
Yeah, I'm trying. I can't share my screen. So I'm just trying to do that. Can anybody see my screen?
No. Sir, just go on up mark or arrow mark.
Yeah, it's just not allowing me to do that. Is it possible for you to reschedule the next presentation now so that I can sort out, because it's not allowing me to share my screen?
Sir, please send the PPT inside the share.
Okay, that's fine.
Just text the email.
Where can I send you?
Yes, I send it. Text box. Email.
Okay. It's a very large file. Just to save time, is it fine if the next speaker speaks now and I can take over that person? So that I can give you the access.
Mr. Clement? Hello, Mr. Clement?
Yes, I'm here. Thank you.
So
Yeah.
So you are here.
Yes.
We would like you to make your presentation right now. Are you ready?
All right. Thank you. So I'm looking also for the option, to upload to share my presentation, and I'm not see that here. So how should you do to share the presentation?
That will be a problem.
I'm looking at that option on my screen, but I'm not see the option of sharing the screen.
Yeah, I have the same problem.
Sir, please click on three dot to get a start setting option for a screen setting.
Okay. Okay, I click on setting. Yes.
Just up arrow mark. Click. Just left, inside left, three dot, arrow mark, up arrow mark. Click
Yes,
on
I
up
see.
arrow mark.
All right.
Just
All right,
click
then I
on the
see.
arrow mark. Just click on the arrow mark.
Up arrow mark, then your next window is open.
No, I don't see the arrow mark there. I see audio, video, general.
Entire window. Your entire screen of window attached with three things. Just click on up arrow mark button option.
Yeah, I click on setting and then I don't see that option that you are giving me. So for some reason. Is it a way that we can email the presentation and you upload?
Please send the PPT in my mail.
So, okay, so I click on setting and I see three options on the left, audio, video, and general.
हाँ पकड़ के लाइए
क्या हो गया? क्या मतलब नहीं कर पा रहा है? क्यों? नहीं कर पा रहा है?
बता इंस्ट्रक्शन दीजिए आप.
Okay.
Sir, I'm KK Shahu. Am I
So,
audible?
to who can we send the presentation to upload? Because we don't have that. I don't have that option here on my screen.
Just click on your entire screen. Just mouse, go over your entire screen and click there. Your mouse cursor is there.
ये हम दिखा रहे थे मतलब ऐसे हो
अच्छा
ये हमारा साइड से
उनका व्हाट्सएप है क्या
मंगवा लीजिए
सैनिक
CCDC office.
half an hour नहीं चार हाँ पहले आ जाएंगे तो अच्छा रहेगा स्टेशन में कुछ ज्ञान कर लेंगे 4 बजे क्या नाम इनका है क्लेमेंट hello Dr. Clement Giorgio.
Yes.
What is the problem coming to you now?
Yes, I'm not still seeing the option, to share my screen.
Do you see the up arrow in your screen? Arrow up or 3 dot there?
Yes.
Just click on the up arrow.
On the right, yes, I click on it.
Then what you are seeing on the screen?
I still see the participant. The thing that I'm seeing are the participant.
Do you see the window or share screen, etc.?
Yes, I see that. Yes.
Okay. Then click on the share screen.
Okay. Yes. Okay, I see.
And then choose the option you are appearing on the screen. You're the opened screen there. Did you open the
I
screen?
think I click on the PowerPoint presentation.
Yes.
Can you see now?
So first you have to open the PowerPoint presentation in the background.
Yes, I have that. I click on it.
Okay. Then share the whole screen that will appear, or the PowerPoint screen was also showing to you.
Okay, let me try again. I think I see that and I click on it on my PowerPoint presentation, but I don't know if you can see it.
Ganesh.
Can you see my PowerPoint presentation?
मुश्किल है समझाना इसको. Can you email your PowerPoint file to me?
Okay, all right, it's Dr. Sahu, right?
sahukk@gmail.com. sahukk.
Okay,
Mail me.
I'm emailing it to you right now, and I also try to CC Dr. Sanjay.
Hi, Dr. Clement.
Good morning.
Good evening for my time. Thanks
All
for.
right, so let me send that to you.
हम मैसेज कर रहे हैं.
इसका भी नहीं हुआ है.
किसका?
डिटेल्स उसका.
उसका वही प्रॉब्लम हो रहा था.
तो शायद आ गया.
किसको भेज दिए हो? गणेश, उसका नितेश का फाइल आ गया पीपीटी. किसको भेजा है वो? तो कहीं भेज दिए हैं देखो.
डिटेल्स इससे पूछ लो ना फिर किसी का हो जाएगा तो
उसका व्हाट्सएप नंबर है क्या?
Okay, my presentation is sent.
प्रेजेंट ऑप्शन खोल ले रहे हैं हम लोग, तो वहाँ जो कुछ जैसे सर ये हम करते हैं जैसे कोई एक ही है, जैसे यहाँ से सेलेक्ट करने के बाद शेयर होता है.
हाँ ठीक
वो
है.
नहीं कर पा रहे हैं, वो सिर्फ वहाँ पर
अच्छा.
यहाँ क्लिक किसी पर सेलेक्ट करें उसके बाद वो शेयर होगा.
हाँ.
ये चीज़ सर
समझाते हैं.
हाँ.
Sir, Clement sir, please try again once more. I am giving you the step-wise instruction. Just click on the up arrow, showing up arrow. Once again, please. Then you will see the share the entire screen.
Yeah, it appears, and I click that. I'm not. It's not doing anything on this time, so I'm not sure why.
And click
But
on the
I emailed you my PowerPoint presentation already, and CC.
Okay, click on that, and there is a button of share. Blue button, share. After selecting your presentation screen. Sanjay ji, are you listening me?
Yes, sir.
Please help Dr. Clement.
Can I share his presentation?
He is unable to present his PPT slide.
No, sir. Can I share his presentation?
Dr. Kumar, Sanjay Kumar, I send you also, I CC you a copy of my presentation. So
Yeah, I try to share myself.
if you can upload it, I don't.
Okay. Just
Sanjay, it is better
yes,
to collect his PPT files to you and you present there, and he will tell about that screen.
Okay, I'm trying.
Sanjay, just a minute. Actually,
Yes.
another speaker is ready. So if you will give the instruction clearly to Dr. Clement, in the meantime, I think that it is better the second speaker will continue now.
Sir, can I share the slide and he can present it?
Okay, why not? Why not? Yes, you just ask.
Okay, just a minute, sir. I'm just sharing. Okay.
रितेश का मिला गया क्या? हाँ मेरे पास आ गया पीपीटी को दे दें. हाँ क्यों आ गया? DDE DBG कहीं देगा से करो. I got your PPT files. I am displaying now. Just wait. फिल कर दिए. मेल चला गया था.
I'm not able to see this slide. So slides. Oh, goodness.
अजय यहीं बैठे हुए हो क्या?
सर इसमें स्लाइड शेयरिंग का ऑप्शन नहीं आ रहा है सर.
नहीं हो जाए, हम कर दे रहे हैं, यहीं से कर रहे हैं.
अच्छा सर.
उनका स्लाइड मिल गया है.
अच्छा ठीक है.
तुरंत कर रहे हैं. संजय जी आ गया है.
हाँ सर.
All right.
Okay, Dr. Clement, go on.
Let me first introduce him.
Okay.
All right.
We are introducing you first. Dr. Clement G. Yet-Jau is working as Associate Professor in the Department of Biological Sciences, College of Science and Technology, Florida A&M University. He completed his PhD in 2006 in Experimental and Health Science from Jackson State University. His research interests in understanding the cellular and molecular mechanisms of therapeutic agents, including natural products and metals in the treatment of leukemia and other malignancies. Dr. Yet-Jau received several honors and awards. One of them is Faculty Scholar Award for Cancer Research, American Association for Cancer Research, held at Orlando, Florida, April 2 to 6, 2011. He has published several research papers in reputed journals. So over to you, Dr. Clement.
Okay.
So, we request you to complete your deliberation in 20 minutes. 20 minutes.
All right. Thank you very much for the kind introduction. And I would like to thank, first of all, the organizer of this important international conference. And also would like to thank my colleague, Dr. Sanjay Kumar, for the kind invitation. And I'm really happy, to be here. And also, it's a great pleasure, to be here. My presentation is about a natural product called Venonia amygdalina deline that I'll be working with, and it should promising for the treatment of prostate cancer. Next, please. Here is the overview of what we discussed this afternoon with you. Next. So a little overview about prostate cancer is that it's one of the common cancer in male, and its incidence keep increasing globally. And also is the second common cause of cancer-related death in male. And about 1 in 7 male will be diagnosed with prostate cancer over the lifetime. And the good news is that only about 3% of male will die from prostate cancer. Next. This disease generally affect male, and also it increase in age, and it's also a racial disparity, so it affect mostly African American in particular, and the mortality rate among African American is 2 to 3 times higher compared to white. And some of the factors that are associated with prostate cancer include diet, obesity, and other factors such as environment and occupation. Next, please. In 2017, we performed some experiment in our lab, and we found that, poor nutrition are really associated with prostate cancer. And study here showing that, poor diet is really associated with a high risk of developing prostate cancer, including tobacco and alcohol intake, as well as lack of physical activities. Next. So since prostate cancer is a major disease that affects many males worldwide, there can be a lot of intervention. And among those interventions, we have systemic and local treatment that can be used to treat prostate cancer. With systemic, we have chemotherapy and hormone therapy. And with local, we have surgery and radiation therapy. So, since all these treatment can be used to treat prostate cancer, what is the problem? The problem is that many of these treatments are really be very unsuccessful, and also life-threatening for the patient. And therefore, new therapeutic treatment are very important. And that is one of the reasons that, I'm doing the research with the natural plants that seem to be not toxic, as well promoting the immune system. Next. So as I mentioned earlier, so if the treatment that I mentioned earlier can be very good, so that patients should not be died from prostate cancer. But on the opposite side, these treatments are very exhausting for the patient, and current chemotherapy techniques have a range of side effects ranging from minor to severe. And we can say that hormone therapy, for instance, will lead to weight gain, erectile dysfunction, decreased muscle mass, etc. So this gives us a opportunity to investigate, the natural product. Next, please. As we already know, many people before us have always said, eat your fruit and vegetable as medicine. And we believe to that because many fruit and vegetable really have a lot of antioxidant associated with anti-cancer activities. And we have been working with this Vernonia amygdalina in our lab, and we actually showing that we have a promising anti-cancer agent for the prostate cancer, and that is actually why we are investigating this. And also, one good thing about the natural product is at the same time, a vegetable that is widely consumed in East and West Africa. Next. Also, a recent study, also in our lab, demonstrate that by taking a high intake of fruit and vegetable, male may reduce the incidence of developing prostate cancer by 75%. And if we take a look at female can also reduce the risk of developing breast cancer by 50%. And the list going on with other cancer as well. Next. To basically, give a boost, for the study that we are doing. We have a cartoon here said that we have exhausted all conventional measures. One last desperate option is to put you on an alternative natural medicine that have a 96% success rate. This is really interesting because we have seen many patients, that they do not really get a complete medical care, and sometimes, when they're at home, using a natural product and home remedy, their cancer is really cured. Next. So our research question is here that does therapeutic doses of Vernonia amygdalina extra inhibit cell growth of prostate cancer known as PC3 cell in vivo in the in vivo model? And if VAD extra inhibits cell growth in vitro for prostate cancer, what is the mechanism by which VAD extra exerts its anti-tumor activity? And our hypothesis is that VA extra inhibit the growth of prostate cancer cell through inhibition of cell growth, cellular growth, induction of DNA damage, cell cycle apoptosis, and apoptosis. And our objective is to determine the molecular mechanism of action of VAD in vitro. Next. So in this research, we look at two mechanisms, the cellular mechanism and the molecular mechanism. And the level of the cellular mechanism, we performed 3 experiments, including the propagen adan assay targeting the nucleus, the MTT assay targeting the mitochondria, and the trypan blue targeting the cell membrane. Next. Here is our data, with the propagen adan, acrama orange, and the MTT. On the top here, bar graph, we see the data of the MTT showing a gradual decrease in cell viability as we increase our doses of VAD. And on the bottom, we see the images of the Akrama orange and Propagium ardens staining. And then in A here, we have the control sample, and you can see that the cell with normal morphology, and between 125 and 500 microgram per milliliter, we see a gradual decrease in the cell viability as well. We can see some apoptotic body as well, cell fragment. And one of the things that is happening here is that the red represents the dead cell, while the green represent the live cell. And we can also see that in the high concentration here, almost all the cells will die because they don't have the normal morphology as well. So they are lost the morphology. Next. The next data show the data of the Trapen blue, showing on the top here the bar graph showing the live cell in green and the dead cell in red. And on the bottom, we have the images of the broad field showing the density of the live cell and the dead cell. And in A, we have live cell, and between B and D, we have the dead cell. And we can see that between B and D, we have a gradual decrease in cell viability as we increase the doses of VAD compared to the control. Next. Next, please. All right, so our next objective was to look at the molecular mechanisms, and this is a systematic, target that we look at, and some of this data have been already published. And in our lab, we are not just testing Xenonia amygdalina deli, but we are also testing for garlic and curcumin. And by the way, one of the students that start working with garlic with Dr. Kumar before he left Jackson State is graduating this summer. Next, please. All right, so we tested, the sulfadylene serine externalization, and here we see we have our data, and we have A, which is our control sample, and B, between B and D, that's our treated sample. And in our control sample, we have 4 quadrants. The quadrant 1 is like the live cells without dead cells. So, and quadrant 2 is like early apoptosis or annexin 5 negative. And quadrant 3 is both necrotic and apoptotic cell, and quadrant 4 is necrotic cell. So this experiment here helped us, to tell us if the cell will die through apoptosis, necrosis, or by both. So based on our data, as you can see here, especially with the high concentration, the cell will very die through necrosis, because if you look at quadrant 4 here, you see that most of the cells are in quadrant 4. And on the bottom, we have the table, showing the increase in dead cell as we increase the concentration of Xenonia amygdalina deli. Next, please. We also take a look at caspase-3 activation, which is executioner of apoptosis. just like in the case of annexin 5 that we tested, we can also see an increase in the caspase 3 positive cell compared to the control. So in this graph here we have, in this graph here we have M1 that represent that represent caspase 3 negative large cells, and also M2 that represent caspase 3 positive. As you can see, as we increase the concentration, there's a reduction in caspase 3 negative and an increase in caspase 3 positive. And the bar graph, the table down here, represent the increase in caspase 3 positive cell compared to the control. Next. We also take a look at nucleosomal DNA fragmentation. In our sample, as you see here, there's actually a presence of fragment in our gel compared to the increase in the fragment in the treated cell compared to the control. Next. So in conclusion, because of time, other data for this study was not presented, but in conclusion, data obtained from the Propagium iodine and the Akuma orange indicate that VAD significantly reduced the viability of large cell in a dose-dependent manner, showing a gradual increase in the loss of viability in VAD treated cell. And similar results were obtained, with the trapen with the N. And flow cytometric data demonstrate that VAD indicate induced phosphoryl externalization and caspase activation in the cell compared to the control. Next. These results suggest that inhibition of cancer cell growth, induction of apoptosis through phosphatidylserine serine externalization, and caspase 3 activation are involved in the therapeutic efficacy of VAD as anti-cancer candidate toward the prevention and or treatment of prostate cancer. Next. For our future study, because this natural product seems to be promising for the treatment of prostate cancer, we should be able to translate the applicability of Venonia amygdalina for treatment of prostate cancer. And what we are trying to do now is to be able to standardize extra of Venonia amygdalina production, and then the development and the stage that it can be used for a clinical trial. Next. These are the students, that work in my lab, and that also, help me, to generate some of the data. And most of this work, was done at Jackson State before I moved to Florida. And thank you very much. I can take a question at this moment.
So thank you. Thank you for a very nice presentation. Thank you. We are in short time, so we are moving to other paper. Thank you once again.
Okay, Dr. Kleibert, it's nice presentation. Thank you.
Thank you too.
Okay, see you.
Our next presenter, Dr. Ritesh Krishna.
Hi, can you see my screen now?
Yes.
Sorry for a bit of drama about the screen sharing.
Ritesh Krishna, let me introduce you. That's your introduction. Dr. Ritesh Krishna leads the computational genomics research at IBM Research UK. Dr. Krishna obtained his PhD in computer science from the University of Warwick and moved to the University of Liverpool for his postdoctoral research, where he was associated with the Institute of Integrative Biology, Center for Genomics Research, and the Institute of Infection and Global Health. Ritesh is an expert interdisciplinary scientist working on the interface of life sciences and computer science. At IBM Research UK, Dr. Krishna is responsible for building scientific programs, collaborations, and strategies that are aligned with the needs of British industry and UK government's industrial strategy guidelines for life sciences sector. So over to Dr. Ritesh Krishna.
Thank you very much. Sorry about the bit of drama about screen.
We request you to finish it in within 30 minutes, please.
Yes, I will endeavor my best to do that. So first of all, thank you to the organization committee for inviting me. It's a real honor to present in front of some of the scientists I have seen from very young age. So today I'm going to talk about the computational approaches to understand soil types. So because this conference is on botany, so there is no need to emphasize the importance of soil. But why soil really is important now is also because soil is connected to several global challenges. First is the food security. By 2050, we are supposed to be running out of food, so we need a sustained production of food. Second is water retention. Soil is the major source for retaining water. And there is one of the global challenges as well. How do we keep our earth sustainable? The third is carbon capture. Soil is the major sink for carbon dioxide. And as we are manipulating soil, including agricultural techniques, we are actually releasing the soil we are releasing the carbon dioxide in the atmosphere. And that is resulting into global warming and all kind of climate crisis, which is being discussed nowadays. Fourth, soil is also important for new material discovery, including the discovery of new antibiotics, because soil contains lots of DNA. It's one of the most biodiverse environments on Earth. So in order to sustain the soil, we need to understand what soil health is. And soil health actually can be seen by different perspectives. One is how the physics-based view of it, how the soil is structured, what is its porosity, how the water flows within. The second aspect is the chemical aspect of it, the chemistry. What is the nitrogen, sulfur, phosphate content in the soil, and how basically they are affecting the soil health. And the third one is the biological aspect of the soil. Soil is home to many species, and their welfare, their conservation, and what they are basically dictates the biology of the soil. And that's where this presentation is focused on. So if we look at the soil food web on the left side, we have these plants which, and the leaves, etc., fall on the ground. They basically create the organic matter, which is consumed by the primary consumers, which are bacteria or fungi, which in turn are eaten by secondary consumers, which are like different nematodes and mites, etc. And then the species become bigger, so spider going down to further to earthworms, ants, and eventually the tadpoles and birds, etc. So we are focused basically on understanding this bacteria and fungi, why they are really important. You can see a cross-section of the soil here in this figure that I'm showing. Near the roots of the plant, there are lots of bacteria which are important uh, actors for maintaining the soil health and the plant health because they are involved in a range of biochemical activities. So if you look at 1 gram of soil, you basically have 10 billion bacterial cells in it. So understanding the genetic composition of the soil is really a mammoth task. Depending on the variety of the soil, the bacterial composition will change. And so will the soil's behavior, soil's health, etc. So that's where the science of metagenomics come into the picture. So in metagenomics, what we do is that we basically take these samples and extract DNA from it. And ATGC, DNA is made of 4 dinucleotides, which can be represented at ATGC, and you get like a whole lot of these ATGC-based DNA contents, and you try to do informatics on that to understand what might be going on in the soil. And that's where the supercomputers come into picture. So these are the supercomputers basically. They look like fancy cupboards with lights, etc. And with hardwares of several million pounds inside. You basically access them remotely through your normal computer and do all your processing here. So these are basically used to ask 2 questions related to metagenomic sample, in this case in soil. So who is there? Taxonomic identification, understanding that which all bacteria, fungi are there, and what are they doing? Functional identification. So, are they actually helping in some of the desirable biological traits like, a proper nitrogen fixation or good intake of ammonia, etc. So we need to understand what are the bacteria there and what basically they are doing biologically, which in turn is affecting the soil health. So this research is basically done by 3 entities: IBM Research, Hartree Supercomputing Center, and Rothamsted Research. Basically about understanding the bacterial population in soil through a high-scale informatics. So to understand soil, let me take you back to the chemical fertilizer industry. So this industry was pretty much born in 1843 when John Lewis developed a superphosphate fertilizer by treating bones and mineral phosphates with sulfuric acid. And he claimed that if you put this mixture in the soil, it would promote plant growth. So this was an inorganic material going into the soil. And he set up this factory, Lewis Manure Factory in England in 1843. And that is really when the chemical fertilizer industry was born. And he recruited a fellow chemist, Joseph Gilbert, to create experimentation so that he could tell his fellow farmers that you need to buy his fertilizer if you want to increase the yield. So in order to do that, these 2 gentlemen created an experiment that is also called as Broad-Buck experiment. It was started in 1843. It's the longest running experiment in human history. It is still continuing after more than 250 years. So the experiment is like this, where Lewis used to live in a village called Harpenden, where Rothamsted Research is located. There was this large patch of land, and they divided this patch into smaller patches, and you can see, there are stripes there. It's wheat growing everywhere, but each patch was treated with different kind of fertilizer. Some patches were not treated at all, so they were like wild types. Some patches were basically not sown anything, so they were like barren lands. And some patches were given nice fertilizer. Some patches were given like manipulated chemical composition. So even though these all patches are in the same environmental condition, growing the same wheat, they all look different. And the reason they look different is because of the chemical that goes into these patches. And that is the demonstration that if you manipulate the soil through inorganic compounds, your yield would be different and the soil health would be different as well. So there's a really gold mine for soil research because there is a long data level regarding not only the content of nitrogen, carbon, etc. in different patches, but now we also have these metagenomics samples. So we now basically know that what is the bacterial composition of these patches and how these bacteria actually might be playing a role in maintaining the physical and chemical health of the patch, which is essentially resulting in the food that is growing there. So for this experiment, we took three types of soil. One is arable soil, the soil which was treated with nice fertilizer. Well, the second was bare fallow, the soil which was not given anything. It was basically barren land, and a grassland. Grassland was again the natural soil growing the natural grass without any perturbation. So you can imagine the arable has lots of fertilizer in it. It is manipulated to increase agriculture practices. The barren land basically is bare, and the grassland is basically untouched wild land basically. So we wanted to see how the bacteria are behaving across three different landscapes and what they actually might be doing, which might give us a bit of insight about what is going on. So I'm here showing the tomography X-ray images of the soil. So if you look at the barren land, which is number one here, you see the soil particles are disintegrated. They are not very coherent. And that's what you see in the deserts as well, the sandy thing, where the water doesn't stay, the soil breaks. Arable has a bit of a structure, but it has very little water. It has some patches of water, but a little patches. Whereas the bare fallows, the natural untouched soil, it has several pockets of water. So let's see what happens when we are looking at the content of it. So connected porosity is how well the soil is connected. And hydraulic conductivity is how well the water can flow within the soil. So if we look at this bare fallow, which is more like a barren land, it is very less connected, very less water. Arable, where we are doing agriculture, is still fine, somewhere in the between. Whereas the third, the grassland, is really good for connectivity and containing the water as well. This is also the figure for the big circles represent how much carbon they contain. So you see where the soil is not touched, there are large carbon patches there. But where the agriculture is happening, the carbon reduces, and in the bare land, there is very little carbon. So the connected land is also helping retain the carbon. That's what this particular figure shows. So what we try to do is that, okay, that was the physics and chemistry of the soil. So let's try to understand what the bacteria and the biology is happening there. So we use bioinformatics and AI basically to understand the metabolic efficiency of the soil. And we generated lots of data, like the genomics data, metagenomics data, which was really large in the size. So 4 terabyte basically compressed data form, and it required a lots of complex computational workflows, which would eventually take over weeks of compute time. So we parallelized some of it and tried to do it within days on our supercomputers to gain some insights of what might be happening. So the first question that who is there in the soil? And there's a schematic of how we see. So if I play it, you will see in the root, if you look at it, bacteria, there are all different kind of bacteria, and these are the classification, how much percentage is there. And you can go down. So if you click further on the bacteria, you can focus on, say, proteobacteria, and you want to further look down who are the members of this proteobacteria and their composition. And it creates a very detailed information about the taxonomic classification of bacteria that are present in that particular soil. So that gives you an idea basically that who is present there. And you can do a whole lot of informatics there as well. If you are interested in a particular kind of bacteria, you can basically focus on those. This is another example of the capability in the soil. So these are three types, soil types that we see: arable, bare fallow, and the grasslands. And we are interested in looking at certain biological functions that are happening. And we want to focus on certain genes which are there in that soil. So these are the name of the genes, that little ones that you see on the top, GSPB, GSPD, these are different genes. So this kind of data basically allows you to really drill down at the gene level, which might be responsible for biological functions, and try to see how they are varying across soil types. So in case, if somebody is interested in developing an organic fertilizer, like we drink yogurt to maintain our gut health, because yogurt has lactobacillus, it creates good bacteria inside our gut. If you want to bring something like that to our soil, treating bacterial composition by inclusion of bacteria rather than, nitrogen, like ammonia, etc. We need to look at what bacteria are missing and how we can promote them. And that's where these kind of insights really help. So this picture is like a system level representation of what is happening. So because this is very confusing, so I want you to focus on the left side, which is a comparison of grassland versus arable. So here you see some of the biological processes like CH4 metabolism, nitrogen metabolism is really high in arable. And why is that? Because this is where the fertilizers come in. Once you put the fertilizer in, these activities spike up, and you see like a larger triangles there. You also see carbohydrate metabolism happening there in grassland compared to these arable. But when you see on the right, which is grassland and bare fallow, where basically no fertilizer is there in either of the two soil types, you still see like CH4 mechanism, but that is smaller compared to what is happening on the left side. But this picture on the right shows that there are a lot more biological activities happening, simply because there are a lot more capability in that soil. So we are not tailoring those soil for a particular purpose. This is where the nature is actually working naturally. Now, the important difference between the two is that on the left side, we are manipulating the soil by inorganic compounds, and we are seeing by introduction of which inorganic compound, which particular biological process is being affected. Because those inorganic compounds are basically eaten by those bacteria and fungi, and they act in certain manners to represent this kind of biological activity that is going on. So the metagenomics also allows you to look at a snapshot across different samples and help you understand where exactly the targeted therapy or a treatment requires in order to manipulate the soil health. And that's where we are progressing further. As we are progressing towards understanding soil health, we are trying to understand the biology of soil in a much better way. Now, soil is very mysterious biologically and very less understood. So before I finish my talk, I will just give you another example of what happens when we collect samples from different places and from different environments, paddy fields, deserts, marine soil, etc. And we collected data from like these 7 locations across the globe, and we looked at phosphohydrolase genes basically in particular. And these on the left, you see like these are the different genes present we are interested in looking at. And on the right, you see different kind of soil types. So you see like a desert in Australia having all these genes where whereas the other, like a shallow estuary or a rice paddy field in China is having only a little of it. So that begs the question, wouldn't you think that a paddy field is much richer in terms of bacterial composition simply because it's more fertile compared to desert? No, not necessarily. Because what this picture is telling you is that there is a conserved uh, biological bacterial ecotype present in all particular kind of soils. What is missing in this picture is that it doesn't show that how much of that abundance in there in desert or in the paddy field. Yes, in paddy field, maybe these genes would be really in abundance, but that doesn't mean that they're actually missing in the desert, which is essentially a proof that nature always keeps its capability. It doesn't necessarily lose it. The capability might be reduced, but it would not necessarily have the complete missing link. So if you try to promote growth of those factors, even in desert, possibly the desert would become a greenland at some point of time. But that's where, these minute observations and understandings are required. Also, microbiomes are hugely surviving species, and that's why, in the Gulf of Mexico, etc., you still see those microbiomes. So the environment not necessarily is changing the microbiome, but is rather changing their abundance. And these are some of the surprising things that we essentially see. So in order to do all this kind of bioinformatics, there is a kind of schematics, especially for the young students who might be interested into this kind of stuff, how the data flows and how the compute happens, etc. Of course, it requires a large amount of compute. That's why IBM Research is involved into that to accelerate some of these things. And with this, I would basically conclude my presentation, take questions. I put names of some of these key technology areas which are emerging, especially for young students to basically look at it. These are all emerging areas making huge impact in biology nowadays. And with this, I would conclude and thank you, everybody.
From your lecture, it is a new approach. Very much thank you. Is there any question? Thank you, Ritesh, for this.
Thank you. If there are any questions, I'm happy.
Combining the computer and the biology, and very interesting. We will try your lecture again after some time as a invited lecture, so that students will be benefited very much. So thank you
Very
very much.
happy.
Any question from the audience? No. Thank you once again for nice presentation. Thank you. Next presenter is Dr. Niharika Sinha. Dr. Niharika Sinha.
Yeah, I can hear you. Hello. Can you hear me?
Okay. Yes, Niharika. Dr.
Okay.
Niharika Sinha. Let us introduce you. Dr. Niharika Sinha completed her PhD in cancer biology from the National Institute of Technology, Rourkela. In 2019, she received USDA, NIFA, AFRI Merit Award from the Society for the Study of Reproduction. Dr. Sinha is working as a postdoc research associate since 2017 at Animal Science Department in the Michigan State University. Currently, she is working on epigenetic reprogramming in the gestational diabetic model. So over to you, Dr. Niharika Sinha.
Thank you very much, Professor Abhaya, for introducing me. And can everyone see my screen?
Yes, yes, yes.
Perfect. Okay. Okay. So, good evening, everyone. And it's good morning here from the United States. So, today I'm going to present developmental programming, prenatal testosterone induced epigenetic modulation, and its effect on gene expression in sheep ovary. So, what is developmental programming? In 1990, British epidemiologist David Barker proposed a hypothesis that when a mother is pregnant and she is under stress, this stress can be of several types, like she can be undernutrition or overnutrition. There are several chemicals, pollutants in the environment. So these stress acts negatively to her growing fetus, and this fetus undergoes fetal reprogramming in different organs. As a result, there is a epigenetic changes in the fetal organs which causes impaired metabolism. So when this fetus grows into an adult, he or she shows metabolic dysfunction. And these metabolic dysfunctions are inherited by subsequent generations. So this study was first done by David Barker, and he saw that when a mother is under stress, the fetus is manifested to cardiovascular disease, stroke, hypertension, type 2 diabetes, and obesity later in life. In this study, we have used a sheep model. This is a Suffolk sheep. This sheep is predefined as PCOS phenotype. PCOS phenotype means they have polycystic ovarian syndrome and they have multifollicular ovary, cystic ovary, which impairs fertility. We have used sheep model because sheep is a precocial mammal. Precocial means when the newborns are born, they are quite developed like humans. And the sheep also has the developmental timeline like humans, and they have as well as metabolic as well as reproductive trajectory similar to the humans. That's the reason why sheep becomes an excellent model to study. Flowchart of the experiment in this project, what we did was we injected sheep 100 milligram of testosterone propionate suspended in cottonseed oil, and they were injected during their gestation at day 30 and day 90. So at day 90, the fetal ovaries were taken out, and some of the moms were allowed to term, and those offspring were allowed to grow for 2 years. And with these two ovaries at fetal day 90 and adult year 2, we did RNA-seq. So you can see a flowchart for RNA-seq here. What happens in RNA-seq is basically you isolate your RNA from your samples, and then those RNAs are truncated into smaller transcripts at 94 degrees Celsius for like 15 minutes. And these truncated RNA then forms the template for your cDNA. So the first strand and second strand of cDNA get synthesized, and your 3' end is adenylated while the 5' end is ligated to an adapter. You can see here is attached to the adapter and adenylated. Now these reads are aligned on the genome. Okay, it's like mapping stuff. So alignment means it's aligned to the ship genome to see how many assembled transcripts we can get. So assembled transcripts, after they have been spliced alignments, it is divided into like up regulation and down regulation of genes. The genes which are more abundant they are upregulated, and the genes which are less abundant, they are downregulated. Now, based on this upregulation and downregulation, heat map is generated. So this heat map gives you an idea about genes which are upregulated, which are in red, you can see here. These are upregulated, and in negative, these are downregulated genes. Similarly, these are the top 5 upregulated genes, and these are top 5 downregulated genes. Similarly, the work was done in adult ovary, and we selected few genes. You can see the red marks in the adult. We selected these genes for the further chip analysis. Before going for chip, we did some bioinformatics analysis. The pathway analysis was done through IPA. It's a software which shows you which molecular pathway is upregulated and downregulated. So in our case, we were more interested into the androgen biosynthesis. As you can see, we are injecting testosterone into the sheep, and which results in a polycystic ovarian syndrome in the offspring. So when we did the gene ontology analysis through David, we saw most of the genes which were upregulated or downregulated, they showed certain biological process like phospholipid metabolic process, regulation of blood pressure. Here, the main focus was the sterol synthesis, lipid synthesis, glucocorticoid synthesis, and we positively see the upregulation of this molecular pathway. So what is epigenetic and epigenetic modifications? Epigenetic is basically the study of gene expression that is based on your behavioral and environmental change. And epigenetic modification is that when in an eukaryotic cell, the genome is packed in a chromatin, right? And these chromatin is the fundamental unit of chromatin is the nucleosome. You can see here a octamer histone proteins that is wrapped around a DNA. And these are the marks. You can see the lysine residues that are either methylated, acetylated. When they are methylated, like H3K9Me3, H3K27Me3, so these you can see the compaction of the DNA. And this compaction means that it is not available for your transcription factors to bind, your enzymes to bind, okay? And when you see H3K27AC, these are the positive marks. H3K4Me3, H3K27AC, these are the positive marks that bind to your DNA, and the compaction decreases. You can see the DNA is now available for your polymerase, for enzymes, for your transcription factors to bind, and initiates and elongates the further process. So based on this idea, we chose some epigenetic marks, like these are the negative marks. You can see gene repressive means it represses gene expression. So you can see we did Western blot for this. And when you compare with control and treatment, you see no difference. Histone is the housekeeping gene. So when we compare in the fetal day 90 ovary, we didn't find any change. But when we saw no change in fetal gene inducing histone marks as well. So in fetus, though it's proposed that there can be epigenetic reprogramming, we saw no change in epigenetic marks. Moreover, when we did a chip analysis with these genes that I talked in the first slide, we selected some genes for the epigenetic marks, and we did in adult, and we, hello. Yeah. So when we did chip analysis, with those genes that were selected from the RNA-seq, we saw a high upregulation of SERPIN5, HSD3B1, CTLA4, HSD17B14, SPP1, OXTR genes on the positive mark. So when you say upregulation, that means the activating marks were upregulated. So these are all positive marks, and they allow your transcription factors and enzymes to bind to your DNA, which results in your upregulation of genes. Moreover, we did a vice versa. We also saw a negative mark expression on the upregulated genes, and as you can see, they were downregulated, of course. And we also saw some downregulated genes, H3K9ME3 negative mark. So these genes are very much important in the follicle formation for the corpus luteum, and it plays an important role in folliculogenesis. So this is a nutshell of all my research, what I did. I would be thankful to my mentor, Dr. Aritra Sen, my lab members, NIH, for providing this grant, University of Michigan, because this was done in collaboration with University of Michigan. And I would like to especially thanks Dr. Abhay Kumar for inviting me to this conference. And at the last but not the least, my family members who have been always a great support and inspiration for me. Thank you.
Thank you, Dr. Niharika Sinhaji, for your nice presentation, exhaustive presentation. The paper is open for discussion. If there is any question from the audience.
Yeah.
So if not, we thank you very much. Thank you.
Okay.
Thank you.
Thank you. Thank you.
जरा सा पेपर देखो तो दूसरी मकान
Now the valedictory session will start. Let's start now. And the chief guest of valedictory session will be Dr. Dolly Sinha, Honorable Pro Vice Chancellor. So all the participants who are present online, please remain for this valedictory session.
Now we are at concluding session of this two-day seminar. So Vice Chancellor ma'am has arrived. Now we are going to start this session with. Now I request to HOD sir for bouquet presentation to the honorable man. I request coordinator of this seminar, Dr. K.K. Sir, and organizing secretary of this seminar, Dr. Karan Prasad, kindly join the dais with our HOD sir and patron. Now I request to Professor Abhay Kumar sir for welcome address. Good evening to you all. Good evening to you all. I welcome all the participants, offline, online, the persons and the delegates from India and abroad in the valedictory session of the two-day international conference on the topic innovative research in the life science or sustainable development. First of all, I welcome our Honorable Vice Chancellor Dolly Sinha due to her constant I don't say pressure, but due to our constant suggestions, we have organized this national in the month of January and the second international conference in this month on 29th and 30th July. So, ma'am, thanks to you that your support, with the support, advice, we have organized this successful seminar, this successful conference. I welcome all our colleague teachers, Professor K.K. Sahu, Professor Arun Jha, and Professor Gajendra Prasad, who helped in every respect in organizing this international conference. Without their support, it was completely impossible. So I thank to them also. I welcome Professor Pushpam Narayan, Dean, Faculty of Fine Arts, Dr. Prem Kumar Prasad, Principal, CM Science College, Dr. Vidanath Jha, retired principal, MLSM College, and Professor S.N. Chaudhary, retired professor of this department. I'm very much thankful to Professor Chaudhary, who constantly helped us, advised us, and supported us in organizing this seminar. I welcome all the students, teachers of different colleges, research scholars, and all the delegates and all from India as well as abroad, who constantly supported us, helped us, and consented for their deliberations. So, thank you all the persons present here, online as well as offline. So, I thanks again and welcome all even the print media and electronic media who has covered the story of all the events right from early in the morning on 29th to up till now. So altogether 20 presentation has been given by different scientists of India as well as abroad. And one thing very remarkable, two of the speakers have shown their willingness for MOU, and that will be elaborated by Professor K.K. Sahu. So it is one of the wonderful findings of this international seminar that it might be possible that one day we can sign an MOU with the government of Germany and other from the government of USA. So it is really very interesting and the outcome of this conference. So thank you very much.
Now I request to Dr. R.N. kindly join the line. Thank you. Moving on, I request to Gajan Prasad sir to represent conference report of today's international seminar.
Honorable Professor Saiwa, respected head of the department, Professor A. Kumar, coordinator of this seminar, Dr. K.K. Sahu, co-coordinator of the seminar, Dr. R.N. Jha, respected principal for various colleges, professor and head, home science, dramatics, colleagues, retired Professor S.N. Chaudhary, come guardian of the departments in various corners. My students, first semester and first semester, and researcher scholar, some media Karmi, technical support by Ganesh Mahato, and technical supports also given by Mr. S.K. Singh, Kumar Singh. Today, I'm going to conference reports, recommendation and outcome of the International Conference on Innovative Research in Life Science for Sustainable Development. Today, outcome of the conference,
three proposal has been comes from for LNMU, BC Sandeep University for agro management in agriculture, bioinformatics proposal by Mathana Genome, Dr. Alok Arun from USA, and lastly third, Dr. Harendra Mahato from Germany, exchange program for genomics and Indian Australian collaboration work. The most outcome of this conference has come in light on the exchange of genomic work, Mathana work, agriculture, hydro or agriculture management. More in various further helpful to the our students of this university. Now come on section 1 per day, part 1 moderator and chairperson Professor Santosh Kumar, retired professor and head University Department of Botany, Bihar University, Muzaffarpur. Speakers Dr. Neha Garg, Assistant Professor, Department of Medicinal Chemistry, IMA BHU. Dr. Ayan Mishra, retired University Professor and Head Department of Botany, LNMU Darbhanga. Ms. Ishita Bajaj, Federal University Berlin. Galen Center for, of Plant Science, Institute of Biology, Applied Genetics, Albert Serbeck, 6 B14195, Berlin, Germany. Dr. Basudev Jambre, MD, Aesthetic Eco Research Private Limited, Nashik. Reporter Dr. Rajajit, Assistant Professor, GD College, Begusarai. Abstracts of the first session. The session focused on the area of research in life sciences, molecular biology, biotechnology, bioinformatics, genetics, biodiversity, agriculture, and healthcare and environment, with a special emphasis on sustainable development. These disciplines of life sciences are the outcome of our vast knowledge generated from research and innovation, and it is a continuous process which is going on. The life sciences embrace a great era of intellectual activity, a continuum extending from origin of life to understanding of total ecology of planet Earth. Presentation by the planet Dr. Neha Garg, Assistant Professor, Department of Medicine Chemistry, IMA BHU. केवल नाम Dr. I.M. Mishra, retired University Professor and Head, Department of Botany, LNMU, Darbhanga. Dr. Mishra delivered an interesting talk on innovative research in life science for sustainable development. Ms. Ishita Bajaj, Dr. Basudev Jambre. In second session, moderator and chairperson Professor Santosh Kumar, retired professor and head University Department of Botany, BR University, Muzaffarpur. Speakers Dr. Eric Bangkam Radloff, Dr. Kaushik Shaha, Dr. Alok Arun, Dr. Harendra Mahato. Reporter Dr. Rajajit. Presenting by the panelists Dr. Eric Bangkam Radloff, Dr. Kaushik Shaha, Dr. Alok Arun, Dr. Harendra Mahato. Conclusion: in their concluding remarks, the panelists noted that available studies are remarkable in their field. The lively debate at the end of the session and follow-up talks after the session between participants and panelists showed the need to improve understanding of the topic involved. Second day in first session, moderator chairperson Professor V.N. Jha, retired professor, Department of Botany, Emulation College, Darbhanga. Speakers like Dr. Krishna Prakash, second Professor Santosh Kumar, thirdly Professor A. Basampian. Reporters Khalid Anwar, Assistant Professor, CM Science College, Darbhanga, Bihar. Presented by the panelists of the session one of the second day. Yes, speaker Dr. Krishna Prakash, second Professor Santosh Kumar, third session moderated and chairperson Professor T.N. Jha, retired Professor, Department of Botany, LNMU. Speaker Professor A. Vaishnavan, Emeritus Professor and IQAC member, BHU. Reporter Dr. A. Anwar, Assistant Professor, CM Science College, Darbhanga, Bihar. Session second day. Moderator and Chairperson, Professor S.N. Chaudhary, retired Professor, Department of Botany, LNMU, Darbhanga. Speaker Dr. Naveen Enam, President, Biotechnology Society of Nepal. Dr. Jampad Kyar, Associate Professor, Department of Anatomy. Dr. Javed Nahar, Associate Professor, Department of Genetics and Plant Breeding, Bangladesh, Agricultural University, Main Singh, Bangladesh. Reported Dr. Ankit Kumar Singh, Assistant Professor, Marwari College, Darbhanga, Bihar. Presented by the panelist session 2 of second day. First speaker Naveen Enam, President, Biotechnology Society of Nepal. Second, Janpat Disanta, Associate Professor, Department of Anatomy. Third, speaker of this session was Dr. Jabatun Nahar, Associate Professor, Department of Genetics and Plant Breeding, Bangladesh, Agricultural University, Nimamja Singh, Bangladesh. Our topic was protein changes in sweet cherry flower bud during dormancy upon climate changes. Third session in second day, moderator cum chairperson, Professor B.N. Jha, retired Professor, retired Professor, Department of Botany, Emanation College, Darbhanga. Speaker Professor Purmila Pachak, Department of Botany, Punjab University, Punjab. Dr. Ritesh Krishna, IBM Research Artery Center, SIFC Laboratory, Warrington, England. Dr. Clement G. Yadjo, Assistant Professor, Department of Biological Sciences and Technology, AM University, Florida. Dr. Niharika Sinha, Department of Animal Science, Michigan State University, Michigan, USA. Reporter Khalid Anwar, Assistant Professor, Science College, Darbhanga. Presented by second day, Professor Pramila Pathak, Department of Botany, Punjab University, Punjab. Second, Ritesh Krishna, IBM Research Artery Center, SIFC Laboratory, Warrington, England. His bright topic was computational approaches to understand biodiversity in soil types. Reporter Ankit Kumar Singh, Assistant Professor, Marwari College, Darbhanga, Bihar. The third speaker of this session, Dr. Clement G. Yadja, Assistant Professor, Department of Biological Sciences and Technology, AM University, Florida. His topic was Vernonia Galadilla jelly, a medicine for prostate cancer. Prostate cancer is worldwide cancer topic governing high value impact always. Fourth speaker Dr. Niharika Sinha, Department of Animal Science, Michigan State University, Michigan, USA. Our topic was developing programming parental modulation and its effects on gene expression, seed ovary. Conclusion. In their concluding remarks, the panelists noted that available studies are remarkable in their field. Their lively debate at the end of the session and follow-up talks after the session between participants and panelists showed the need to improve understanding of the topics improved. Final session third, second, oral or poster presentation. Chairman for presentation, Professor S.N. Chaudhary, retired Professor, University Department of Botany and LNMU, Darbhanga. Reporter Dr. Rohit Kumar, Bihar University, Muzaffarpur, Bihar. There was a sum total of 18 oral presentations from different fields of life sciences. And 3-4 poster presentation judged by poster presentation Professor K.K. Sahu and Dr. Gajendra Prasad, the Department of Botany and LNMU, Darbhanga. That is the last or all the themes of the international conference. Thank you very much.
Thank you, sir, for sharing all the details regarding today's international seminar. Now, moving on, we would like to hear some feedback from the side of participants. So, I request one feedback from teacher, another from PGA scholar, sorry, PhD scholar, and another from PGA student. So kindly raise your hand and join to us for feedback. So we have Khalid Anwar sir from CN Science College.
Very warm evening to one and all present here in BBC and all the delegates. So on behalf of the faculty side, in the form of feedback, I'm here to represent my opinion. Well, I've attended so many conferences here and there, national as well as international, but this is the first time in my life attending a seminar or conference which is in the hybrid mode, that is online as well as offline. Obviously, every act has some pros and some cons. So here we also face that, but the experience we gathered here is awesome. It's very delightful and glad to be meet. So overall, the experience is very good. However, certain technical issues may happen because one is sitting in front of us and another miles away from here. So there might be certain sound problems or other such type of issues, but that's not in the vicinity of our effort. So overall, what I can experience and share along with you all is that it was very good, informative, delightful, and impressive talks. Moreover, I can add that the talks was broad and bright because it began with molecular biology, touching to the fields of physiology, and thereafter the clinical biochemistry also. It described the different talk about the cancer treatments, the different type of transgenicity in different plants, the mutations, and overall, in some few words, we can describe that whatever we can share for the betterment of human life and for the betterment of the survivors. With all these words, I end my views and thanks the organizing committee for giving me a chance to share my words. Thank you very much.
Thank you, sir, for your brief feedback. Now I request from PhD scholars, please raise your hand and kindly join us for feedback. Anyone, please? So from the okay, if any person who has joined us for online, so one student who is interested to give us feedback, so kindly join us.
Respected guests, our HOD, and all the members, participants, my colleagues.
and all the students. I am Ajit Kumar, a researcher scholar in the Department of Botany at LNMU University. In my opinion, the two-day international conference, the seminar was a big move, and the seminar going very well, and my experience was very excellent, and thank you very much.
Now I request to Ujjwal, a student of third semester, he wants to share feedback with us. Kindly join Ujjwal.
Respected delegates members, I am Arjun Kumar from MSc Botany Department. In expressing my feeling about the World Conference, first of all I want to thank my respected professors, Professor Abhay Kumar, Sahu sir, Professor Arindra sir, and our Professor Vicky sir, as they provided us the chance to think over how to think in the research field, and presented and provided us a moment in which we meet to such a great delegate personality and being able to take certain new frames of thought through which we can see the world. In the following conference, we will introduce with the real meaning of sustainability and its social, economic, and cultural views. Later on, we will in this conference introduce with several topics such as the cancer cells by Dr. Neha Jain, about the coronavirus and the new disease. These are too much helpful to us, and we found it that if this type of conference may go along, then it will be too much beneficial to us. And we wish that in the future this type of conference will continue so that we can get shower of such blessings. Thank you.
Thank you, Ujjwal. Now I request to our patron and provide compliment for valedictory address. Ma'am, kindly join us.
सभी को मेरा नमस्कार and very good evening. Respected personalities on the dais and in the audience, we have with us Professor Abhay Kumar, Head Department of Botany and President of the conference. We have our delegates and dignitaries who have joined us through online mode, participants, faculty members of botany of the university department and also from various colleges of the university, and distinguished academicians and personalities who I can find in the audience: Dr. Pushpam, Dean Faculty of Fine Arts; Dr. Prem Kumar Jha, Principal, CM Science College; Dr. Vidyanand Jha, retired Professor of Botany; Dr. Chaudhary, retired Professor of Botany, and all members of the organizing committee: Dr. R.N. Jha, convener; Dr. Rajendra Prasad, organizing secretary and treasurer; Professor K.K. Sahu, representatives of press and electronic media, and my dear students. It is indeed a matter of great happiness that the Department of Botany, LNMU University, has successfully, I emphasize the word successfully, conducted a two-day international conference titled Innovative Research innovative research in life sciences for sustainable development. The conference was conducted in hybrid mode, and that is the power of modern science. Through this mode, we can reach to large number of people, and I do not know the exact number, but it must be more than 500. What is the exact number? I don't know. 400. So I guess it would be 500. So it is exactly 400 something. So that delighted me that the department has conducted the international conference in true spirit. Even this is the last session, and I'm happy to see that all people associated with this conference is very serious. They are not taking this last session lightly, which usually happens during uh, most of the conferences. I also attend many conferences, but after the technical sessions, people start taking things lightly, but which is not happening here. So for that, I congratulate the department and the organizing committee, people, the researcher scholars, the supporting staff members who all have assisted behind the scene for successful conduction of this conference. The list of resource persons and the academicians who have chaired the sessions, panelists, and also presenters is very, very impressive. I came to know from the report that scientists, academicians, and people from industries from various countries, including USA, Sweden, Bangladesh, Sri Lanka, Nepal, and other countries, Germany, many people are there from Germany had joined the conference organized by LNMU universities. I am very grateful to all the delegates who have joined us and gave their precise, valuable time for sharing the knowledge with our teachers and students. As we all know, the 21st century can become as the century for life sciences. Earlier, we know that 20th century, 19th century was for physics, then 20th century there was lots of development in applications of physics like lasers, and microscopy has developed a lot in 20th century, when it was also there was a lot of development in chemical sciences. But this century, I feel, is precisely for biological and life sciences. So research in biology is very, very important for the well-being of human beings as well as all other living things on this planet. And the development in life sciences is all because now the technology has given us the power so that we can manipulate at the molecular level. That is the beauty of the science these days. Manipulation at the level of molecule is possible now, and through that we can fabricate biological machines also to our desire. So this is very important with the support of various types of basic sciences, physical science, chemical science, and technology involving computer science, artificial intelligence, and machine learning. The biological, now we call biology as biological engineering. The branch of biological engineering is developing like anything. It is exponentially developing, and it has tremendous impact on the economy also. All business houses are eager for innovative research breakthroughs. They are taken by the industries very quickly, and also manufactured things are manufactured and it comes very soon to the market. So there is lots of potentiality. I am very happy that the department department has organized this type of conference, and from the report you knew that various areas, modern areas were touched upon, and I'm very sure that my dear students, my students, you have enjoyed your 2 day conference here very much, and you are also academically enriched. I think the purpose of organizing such conferences is to share knowledge, and if the goal is that, and if the department has made a breakthrough in that, that is your achievement. So, from the face of my students, I can find out that this is the last session, but they are not in a hurry to leave. So they have really enjoyed the 2 day conference. Regarding sustainability, I would say, previous day also, our Honorable Vice Chancellor has mentioned that there are 17 goals, and out of these 17 goals, the 8 goals are directly involved with biology. The first goal, if you remember it, is zero hunger. And that is the Department of Botany. No other department can have more access to that than this. Zero hunger. Who can solve this? We want to achieve the target of zero hunger by the end of this 2030. That is the goal, that we want to make hunger zero for the whole world. And hunger, when we say, it is also involves hidden hunger. That is the need of protein and food. I am not a person of biology or medical science, but hidden hunger means it is related to the intake of protein and the need of protein for everybody. So who can solve these problems? It is you, the persons who are studying botany and agriculture. The second goal is good health and well-being, and there also biology is directly involved. The third, clean water and sanitation. The fourth one, affordable and clean energy. 6, climate action. 7, life below water. 8, life on land. So life on land, life below water, this is all related to botany and agriculture. So all that, out of the 17 goals, 8 goals are directly related to life sciences, and other goals are also indirectly related to life sciences. So what we want on this earth is the well-being of human beings. And not only human beings, along with people want to be good, I want to have a comfortable life, a happy life, then everybody around us should be happy. So in Sanskrit we say Sarve Bhavantu Sukhina. सब लोग एक साथ सुखी हो सकते हैं. So there is the importance of environment, biodiversity, everything is important, and with the level of that we have achieved nowadays, and the knowledge of basic sciences and technology also is there in our hand. So if we apply your mind, you can also innovate great things. So it is not that only the scientists working in very good laboratories can innovate. Even the students nowadays, the students of various universities and institutions, they are coming with great ideas. And if you have ideas, we also know that in India also now you have various scopes like Atal Incubation Facility and other facilities are there. There are various institutes which helps you in growing your ideas in actual application level. Like KIT in Bhubaneswar, they also help. We can have an MOU with them. We can invite them. They can also help us in developing the ideas brought by students into actual projects. So once we invited them in Patna University, and they have selected 3 ideas of the students, and one when she was a girl, a student, she has a project. Immediately, they announced that they will support the project with 10 lakhs rupees for its development. So, my dear students, there is lots of things you can do, and you should be grateful that your teachers, they are so skilled, they are so academically oriented that they are doing all these things. Why they are doing? All of them have become professors, and in their academic or career graph, professor is the highest achievement. So they are not going to achieve anything. All they are doing, it is for the development or the growth of you the student. So I think you have enjoyed it about uh, zero hunger or hidden hunger. I attach maximum emphasis to that. We have to think about that, and to our um, satisfaction, I came to that. I came to know that even in the lab now we can grow meat, and I think that meat will be synthesized not through the geological part but through the botanical part. I think so. So that can such type of things also can be done. So how these things are done, we can do. We can go to the laboratory. You attend classes regularly. Go to the laboratory and do experiments. And your subject is so majestic. It is, I can call it a majestic and beautiful subject. No other subject can have so much of applications and so much of scope, and also so wide in so broad. Whatever, if you go to the garden, you can do your experiments. Wherever you are, you can do some experiments. Only thing, you have to be very, very observant, and you have to think a lot. You have to study also a lot, because modern science is not that easy. You have to learn the basics. So I always like to communicate with my dear students, so I can see the happiness in your face. But my dear students, if you have really enjoyed, and if you want to come to the department, you want to learn, you want to work, you have to follow the COVID appropriate protocol. I can see that you are not following the protocol. Our Honorable Prime Minister has time and again always appeal to the youth particularly that you follow the COVID protocol. Do not invite the third wave. You have already invited after the first wave diminished. We invited the second wave. 2020 we lost. 2021 also half the year we have lost. And now we don't have any luxury to invite COVID third phase. So my dear students, be careful. We love you very much. All my students, I am telling from my heart that I love my students the most. We don't want to see you unhappy and ill. So take care of yourself, your health, and also take care of others' health. By wearing the mask, you protect, not only protect yourself, but you protect others. So if you are not wearing mask, you are selfish. You will be stamped as selfish. So be careful. I know all of you are very nice persons. You are not selfish at all. So you are responsible citizen of the country. You are PG students. So you must follow the protocol and make awareness among general masses, as well as people younger to you and people who are less knowledgeable than you. So once again, I extend my best wishes and congratulations to the department for arranging such wonderful, wonderful conference. And I think all the good papers that come will be published through a foreign publishers. I wish you can publish it in a foreign, to a foreign publishing house, so that would be very good. And about the MOUs also, I'm very happy that 3 proposals for MOU have come, and I wish we can materialize those things. And in future, we can have student exchange program also. We can send our students abroad. Some students from abroad can come here and work with us. So there is lots of scope here. New Education Policy 2020 also promotes research for undergraduate students right from from undergraduate level, students should do research because teaching learning without research does not have any sense. So you are the new generation. The sky is not the limit for you. You can go beyond the sky. Being a person involved with physics, I will say that there is much, much more space outside the sky. So your sky cannot be the limit for you. You have the whole universe for yourself. So develop yourself and, and take advantage that you have got such wonderful teachers. Thank you. Thank you all.
Thank you, ma'am. Thank you.
Thank you so much, ma'am, for your valedictory address. Now I request for vote of thanks to coordinator of the seminar, Dr. K.K. Sahi.
I am breaking the Corona protocol for a while. Excuse me, ma'am. Honorable Pro Vice Chancellor, an ideal lady, ideal academician, and ideal personalities in all respects. Our head of department, Professor Kumarji, Dean Faculty of Fine Arts, Dr. Krishnan Rayji, my guru, my leader brother, my whole and soul for making the sake of this department, Professor S.H. Officer, Chief Science College Principal, Prem Kumar Prasad, the ex-principal of Medical College, Botany Ji, and ex-principal Virali, teachers from different colleges, students, researchers, scholars, like media, and the person who is online listening to me. It was a great, great, great experience that I have ever experienced in my life. The 2 days that I did my work with the limitation of energy, but I think I did much than that. Actually, the organizing of a simple program to a regional program, to a state level program, to the national program, and managing the international conference is a very, very, very vast. Fortunately, I am very much lucky that we have a team of different types, different categories, different knowledge, different skills, and they all, in different ways, help me in every moment of problem, every moment of the things that I can never, never forget. The vision of this conference, the planning of this conference, managing of this conference, and executing this conference, a lot of steps, lot of things, lot of branches, lot of things to be done with a proper management, with a proper skill, and they all come from my colleagues. They all come from the supporters. I don't like to say all about that to help me. They're all friends, all students, all the third and fourth grade employees with this department and the DO office department. The person who is sitting apart thousands and thousands of kilometers away, they also help me in a various way by managing the staff, managing the different, making the schedules, and even the designing of the staff, etc. I don't and never forget the help that I gathered from them. In this scenario, I don't like to name all of us, that it is a hidden to you all, and it is my heart that is thanking to them in various ways. I don't like to say anything about that. My love to my students, my love to the few of his future and her future, and I am sure they all students must have gathered the knowledge
and experiences, and the vibration must have come to them so that the thinking process towards the research in the botany, thinking process of the helping to the others, thinking process to innovative ideas, and thinking process of sustainability must have come in other aspects of the community. Thank you very much to you all.
Thank you, sir.
Thank you, sir. Thank you very much, sir.
Now it's time for national anthem. I request to Gayatri and her team to please come and join with us for national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उज्जवल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you to all. Now it's over. It is completed.